Yes, thank you very much for the kind introduction. It's really a pleasure here also for me again to resume a little bit the discussion that we had at the ICH level, and I really also appreciate that I see many of the colleagues of the expert group for the ICH and also for the pharmacopoeia working group being here present. So please correct me if I'm saying anything wrong. So the first part will be a little bit resuming what has been stated in the ICH, and then I will give a short overview on the discussion amongst the European regulators also. ICH Q5A was for a long time in place, and the revision came quite late after decades, but of course there are always scientific developments, and that there were a number that what was at that time identified mainly from the initiative, also from US FDA, acknowledging that we have new product types like viral vectors, and we have seen contamination issues with these viral vectors, but there was also the idea also that these some of these viral vectors at least might be amenable to viral clearance method. I will not focus on this. I will focus on the second topic because this is a conference about NGS. And of course, we were acknowledging there have been exciting developments with next generation sequencing, but also with other virus testing methods. So this was the big topic. And for me, this was also personally the main reason in going into the revision, because this is a really exciting new development. Of course, there's also there are other things going on in biotechnology, such as continuous manufacturing, which is poses some challenges in the viral safety control strategy. And of course, also a lot of experience has been accumulated with viral clearance steps. So here also the idea is to use this experience in order to streamline a little bit the product-specific validation work. But I will focus only on the topic 2, on the new test method, and specifically what has been or what is being said now in the revised guideline about next generation sequencing. And coming to the section that describes the testing of the master cell bank, of course, the philosophy of the ICH is really first to have a thorough testing on the cell bank, because this really defines the risk, what is in, what is not in. It defines a little bit the clearance strategy, not only as, of course, the whole production process and other contamination sources, but really the analysis of the cell substrate of the cell banks is an important factor. And this is why the ICH says, as it said before, testing for adventitious viruses should include both broad and specific virus detection assays. And you know that this broad specific assay, traditionally it has been the in vivo assay and the in vitro cell culture assay using indicator cells. But here now there's a clear statement that's saying introduction of new methodologies such as NGS for detecting a broad range of adventitious viruses is encouraged. And this is the specific table. And also here I try to it's a little bit, yes, you have to read very carefully the small printed footnotes in this table. And here, but I try to highlight it in red font. It's really said for that non-targeted NGS can replace the in vivo assay and supplement or replace the in vitro assay. So both can be, the wording is a little bit broader for the in vitro assay than for the in vivo assay. You may notice this phrasing. And it also says that also that NGS can also be used to replace also the assays like the antibody production assay, the MAP, HAP hamster antibody production or rat antibody production assay, which are per se also in vivo assay where some of the test item is inoculated into the rodents, and then people are looking with antibodies whether they had been infected or not. But it's an in vivo assay, of course. So it clearly opens, and really, why to replace the traditional assays? They do have some limitations, certainly. This is just a short outline, yes, of the in vivo assay. On the one side, using rodents, hamsters, sometimes guinea pigs, the suckling mice, where the test item is inoculated into various parts of the animal body, including even the brain. And also we have the inoculation into eggs and embryonated eggs. And certainly the in vivo assay has some list of limitations. The list is probably not totally exhaustive. I see this is as a really a little bit traditional, or of course, in the early days when cell culture were not that prominent, people had to use viruses to propagate viruses, and also decades ago people were more often using also animal as for production of medicinal products, just remembering the monoclonal antibodies that were produced in ascites. And then there might be some non-laboratory adapted strains in this in vivo derived materials, and that probably might not that good to be seen in the in vitro cell culture, and this might have been historically some argument to using this in vivo assay. On the other side, there is a lot of limitations. We have to look in the mice for clinical signs. Not every virus produces clinical signs. That there's a lot of things that can go wrong. Death by multiple cases, like the injection trauma, bacterial contamination. The suckling mice are sometimes cannibalized. Some media components are severely toxic, specifically in the vector production, when people have to add a triton or detergent for cell lysis, and then injecting this stuff into a mouse brain will kill the mouse per se. Yes, of course, the viruses, sometimes we have replicating viruses that interfere with the virus detection. There is often a limited sensitivity. Karin yesterday presented this comparison with the in vivo assay. For a small number, the sensitivity is good, but for many strains, the sensitivity was poor. And of course, it's using animals, and we want to get rid of animal assays. So this is what the ICH now says specifically for the in vivo assay. Non-targeted NGS is encouraged as a replacement for in vivo assay due to its breadth and sensitivity for virus detection and the limitations of the in vivo assay. Further, this promotes the global initiative to replace, reduce, and refine the use of animals. So for me, it's a clear message to go forward in replacing the in vivo assay with NGS. Coming back also to the in vitro cell culture assay, of course, this is also a well accepted assay, and of many contaminations have been detected. It's not that easy. It has been standardized, but it takes a long time, which is sometimes some limiting factor, specifically for the unprocessed bulk release when we want to work as quick as possible. And also here, the things are also not always that golden, and it might look for some people. Of course, that there is some advantage or some good experience. Let's say that the advantage is one can oculate in many cases, not in all cases, a quite high volume number on the high monolayer, on a high amount of monolayers. So this is a good factor in increasing the sensitivity. And certainly also that there are some lab strains around that have been well adapted to cell culture, and they might cause some nasty cross-contaminations in the facility. And specifically, those tests can be well, of course, those viruses, strains can be well detected with these assays. On the other hand, looking on viruses that come from animal-derived raw materials, these strains are not that well cell culture adapted, and we learned this, for example, with the murine parvovirus, the MVM. So there's a well-adapted lab strains, but with the contamination events, these strains were not that well adapted, and they did not produce a pronounced cytopathogenic effect, and they were not that easy to detect, and the sensitivity was orders of magnitude lower. So doing only the comparison with the well-adapted cell culture strain does not really reflect the whole contamination scenario. And so as I said, yes, the limitations are also, this is a long list, probably not also exhaustive. Cell cultures are specific for a range of viruses. Not all viruses introduce cytopathogenic effect. We have to be careful with the transport. We need to have a well-trained personnel. And of course, there's also the interference, because it's a contamination. It's a living system, the indicator cells, and many components can interfere with the detection of the virus, and sometimes even the product itself can interfere. And so even though it's a compendial assay, and compendial, this is my understanding, you have a cooking recipe and you're doing, and then everything what works well. But giving the same cooking recipe to someone who does not know how to cook, probably the result might be very bad. So compendial really does not say that the quality is per se always good or excellent. Just I want to remind also the in vitro assay has its limitations, and that's why also the idea came into the ICH also, that there are advantages with the NGS. So NGS or other methods may be used to supplement or replace the assay. This could address general limitations of the in vitro cell culture assay. And so we tried even to outline some of these limitations, e.g. the susceptibility of cell lines to infection and specific limitations of the production system, so tested mediated interference or toxicity. So how to validate? So there are some hints in the, or some basic principle on the validation. So the ICH guidelines says that we should use suitable reference methods and a panel of viruses of distinct physical properties. So we try at least mainly for the wet part to reflect all the capsid structure for the extraction and all the genome structure that are crucial for the library construction. And then it says also a comprehensive viral database should be used. Of course, what is not in the database will never get out, and of course everything that it's false annotated will get put out. And we were not that specific on the types. So other types of reference materials may be used to evaluate the specific technical and bioinformatic steps. So we are also acknowledging, and we come probably later with the pharmacopoeia chapter, that on the one hand you do an end-to-end validation, but on the other hand it's certainly, at least for the qualification phase and for the establishment, really you have the good control on each sub-step, let's say the extraction, the sequencing, the library construction, and so on. And the validation/qualification includes testing of the general assay performance and matrix-specific validation. So we are always differentiating between a general assay performance, looking what viruses can be picked up with this assay, with what sensitivity. And then, of course, this is a part of every validation, that sometimes we have this nasty matrix-dependent interfering effects. So also a matrix-specific verification would be used. The basic that we also stated here, NGS is a qualitative test, a so-called limit test in the ICH world. So it means we want to have at validation a definition of the sensitivity of the limit of detection, but also on the specificity, which is sometimes also regarded on the breadth of detection. Of course, specificity means you don't want to have a false positive, but it also means you want to pick up a virus and nothing else, but you want to pick up also all viruses. I had also some discussion. I'm also learning, because I'm not a QM person, between validation and qualification. So method validation requires predefined performance criteria. So for me, it's some kind also of verification. So validation is not science. It's not reinventing the wheel, but it's verifying that the wheel really works as it should be. And qualification only evaluates the performance of the characteristics of the method. And here there are some hints. So for any NGS method, validation/qualification should be provided to support the intended use for applications. This includes method validation and matrix-specific verification when used as a replacement. So the basic strategy is when you want to replace a method, it should be validated. On the other hand, we are not that strict with when we want to supplement it, then it's only called qualification. And now how to replace? Of course, this is the question now, how to proceed. So here also the It said that NGS, non-targetitious NGS can replace the in vivo test without a head-to-head comparison. We also discussed it in the pre-workshop yesterday, so there is no exact head-to-head comparison required. And it also says the guideline that non-targetitious NGS may also used without head-to-head comparison to supplement or replace the in vitro. So for both the in vitro and the in vitro, no head-to-head, no strict head-to-head comparison is required. And why I think this makes sense. And of course, we have two different systems, or two systems, different methods. And yes, for me, it's sometimes a little bit comparing apple with pears, and the in vitro assay, let's say the apples, it's a well-established assay that really picks up some strains with very excellent sensitivity, but of course the breadth of detection is limited. On the other hand, the alternative method, it's by the definition might detect all viral nucleosides, or at least even new viruses and so on. And here, of course, the discussion is also about the sensitivity. Is it that high or not? And I think I'm also happy to learn here more about the sensitivity in the comparison. But this is, of course, always with the regulators, yes, there's some kind of consideration. Is it sensitive enough or not? Let me just summarize again. Yes, so the main messages that the ICH Q5A gives, the revised, it really encourages NGS for testing of cell banks, because I think with cell banks also for me, the sensitivity is not, usually we have a contaminant that has been grown for several passages for adapting and so on. At least the cases I have seen, there was a quite substantial amount of contaminant always in the cell cultures. So the utmost sensitivity for cell bank testing is probably not that big issues. The ICH encourages really to replace the in vivo test with NGS without head-to-head comparison. It allows also to replace, or encourages also to replace the in vivo antibody production test, either with specific PCRs or with NGS. I think both methods are equally considered suitable. And it also allows to supplement or replace the in vitro cell culture assay without head-to-head comparison. This has been clearly stated. And as I explained before, method validation is expected for replacement, while method qualification is expected for supplementing the in vitro assay. And of course, as to differentiate, the method validation needs predefined criteria. So these are the method of ICH. Let me now a little bit also resume the discussion that I'm having with my European regulatory colleagues, either in the context of discussion, the Biologics Working Party, also on. So here I have the clear statement disclaimer on the slide. This is not no ICH regulation anymore, but I just resuming, I try to pick up a little bit the discussion with that we're having, and yes, so we are considering both the generic and the matrix validation of NGS methods is performed using a set of properly justified model viruses, and we will hear this afternoon about the reference materials, and certainly the WHO panel is certainly a carefully selected panel. But on the other hand, sometimes we don't want to keep it that strict. So if probably justify one or several of these viruses may be replaced, or other model viruses might be used. So these were just practical examples that we have, because the panel has not been yet that widely distributed. Of course, I'm a really fan of the WHO panel. But we see sometimes the limitations in getting these panels. And there's no head-to-head comparison. This is just a repetition of what has been stated. And of course, it's important that NGS method is capable to detect the model viruses with sufficient high sensitivity. So in this context, it might be useful to compare the sensitivity with the in vivo assay or on, and as Laurent also discussed already yesterday, but this really does not mean need to be an exact head-to-head comparison. And also we have the clear statement that the so-called non-inferiority may not be necessary in each case for each virus. There might be difference. For example, influenza viruses is well picked up with an in vivo vitro assay on the other hand, having a cell substrate that won't replicate anyway the influenza virus. So there might be justification. And we really see the advantage with the breadth of the detection. So we, again, we are encouraging the in vivo testing, certainly. So there's really, I think, a common consensus go forward with replacing the in vivo testing. And also with the in vivo testing, of course, we have been accommodated to the in vitro testing for decades, and many contaminations have been picked up, and it's certainly a worthwhile and a great assay. And on the other hand, yes, here probably that's my feeling that regulators are a little bit more, let's say, cautious, and so what we want to see is a large number of model viruses will be included in the spiking study. So this does not mean that the people need to use the large number of model viruses for each validation or for each product, but it's always good if we have the providing contract lab also if they really have explored the breadth of detection. So a kind of generic or general validation databases that shows, yes, we can pick up 10 or 20 or hundreds of viruses. That's fine, and it really helps to get support in even replacing the in vitro assay. And the selection or number of model viruses for the spiking study should be justified based on a risk assessment. So of course, for not for each product, each class of viruses might be equally needed to be considered. And another argument for including more viruses is also sometimes a little bit of pre-treatment. So for example, if people are doing nuclease pre-treatment, there might be some viruses around where the nuclease works or works less. So that's an argument why we are a little bit cautious in getting the whole panel experience. And again, but matrix-specific validation using the WHO panel or an equivalent panel as justified is expected. So the general theme is to have a broad generic validation, and then doing with a minimum set of model viruses the specific validation. And yes, this is discussion coming up. Yes, the in vitro assay may have an excellent sensitivity for certain cell culture strains. And as I said before, the large volumes can be tested. You never have it. You never have it either with a PCR or with the NGS. On the other hand, the in vitro assay, yes, there are sometimes really severe limitations. For example, replicating vector or replicating vaccine that is difficult to neutralize. Then the sensitivity gets very poor with the in vitro assay. We have seen these cases with pox virus derived vectors. Here the in vitro assay works bad, or let's say vesicular stomatitis derived vaccine. Here also the in vitro assay will not work well. And then the sensitivity really gets poor if you have problems with the neutralization. So there might be really also good justification to replace or abandon the in vitro assay if it does not work. And of course, also no head-to-head comparison is required. It's still recommended to compare at least a little bit the methods. And I think here also, I think also regulator will be getting accommodated more and more, and I really appreciate also to see more and more data comparing a little bit the in vitro assay and NGS. And NGS, if it's properly done, it's really for me a little bit like PCR. You can pick up single or few copies, and also comparing, we have to acknowledge that not each genome means an infectious particles. So again, comparing apple with pears. So usually in cell culture, when we have a contamination ongoing, the cell produces some infectious particles, but usually a surplus of non-infectious genomes, and they will be picked up by the molecular method. So this is a factor in favor also for the sensitivity of the NGS, and really here having a nice comparison will help. And as I said before, doing the comparison only with the utmost best cell culture adapted strain, for me, it's really not fair, simply because some of the non-well adapted strain, they will not be seen that easily in cell culture assay. So this one slide says also we are rediscussing also a little bit the sensitivity. And as I said, for the cell bank testing, probably sensitivity is not that big issues because virus usually grow over several passages. Might be more an issue for the unprocessed bulk testing because here the contamination might be late and you have only a few days or a few weeks for the virus to replicate. On the other hand, also here we are a little bit differentiating between products that undergo viral clearance afterwards, and you could do some kind of this ICH type like risk assessment saying if I anyway have a viral clearance, I also don't need the utmost sensitivity for such products because then the traces amount really might be cleared away. Of course, it depends from product to product, but let's say for monoclonal antibody, we really have broad inactivation methods or removal methods such as the parvovirus filter, which have an exact broad spectrum of removing contaminants. Not all, but most. And so we have more reservation, let's say, a little bit with products that don't undergo clearance. That's like vaccines and viral vectors. That there's more discussion. And here, this is my experience from my colleagues. We still want to have experience how it's performed because it's a new method. And of course, people, when it comes, supplementing is always great. That's clear. Supplementing apple with pears, yes, you have the full spectrum. But when replacing that, then of course the sensitivity is always a matter of debate that I see coming again and again. And here a more cautious strategy is advised. This does not mean that it works. So, and as I said, it's desired to supplement in vitro testing because we have these limitations of the in vitro. But when replacing, then of course we want to see some comparison of the sensitivity and then probably some product specific discussion what we need or what we don't really need. And certainly this might be really product specific because, as I said before, so some products or some vaccines perform very poorly in the traditional in vitro assay. And I would like to thank you for the attention.
Thanks for a great introduction and overview of NGS and some of the regulatory, or I guess thinking also included. So we're not going to have questions after each presentation because we've got an expanded panel discussion after at the end of the session. So now it's my pleasure to introduce my co-chair, Dr. Laurent Mallet, and he's going to co-present with Dr. Gwenelle Surface, and both are from the European Directorate for the Equality of Medicines and Healthcare. And so I welcome you to present the new chapter for the HTS, which everyone has been waiting for some of the details of the validation that's needed, which could not be included in more of the general Q5A format. Thank you.
Thank you for the kind introduction and hello to all. So EDQM, European Pharmacopoeia perspective on next generation sequencing. So in this talk, we are going to cover Council of Europe, EDQM, and the European Pharmacopoeia. Then next generation sequencing in the European Pharmacopoeia. So what is the current situation with NGS in the Pharmacopoeia? And then we'll move to the elaboration of the new European Pharmacopoeia General Chapter on NGS for detection of extraneous agents. And we'll go through the content of the draft documents that recently underwent public consultation. Council of Europe is an intergovernmental organization. It's headquartered in Strasbourg with 46 member states and representing more than 700 million of citizens. EDQM, so that's European Directorate for the Quality of Medicines and Healthcare. So it's a directorate of Council of Europe. It is based on the Convention on the Elaboration of a European Pharmacopoeia. So that's a partial agreement with 39 European member states and the EU. And the mission of the EDQM is to contribute to public health and access to good quality medicines and healthcare in Europe. So an EDQM is responsible for the European Pharmacopoeia. So European Pharmacopoeia is the official pharmacopoeia in Europe. So the aim is to provide quality standards for all medicinal products and their components. And these standards of the pharmacopoeia, they are binding in the 39 member states of the European Pharmacopoeia, and they are also used well beyond, so as a reference worldwide. And there are also 33 observers for all continents that are taking part. So NGS in the European Pharmacopoeia. So since 2017, the use of NGS is foreseen in the European Pharmacopoeia. So that's in the chapter 5.2.3 on cell substrates for the production of vaccines for human use. And then chapter 2.6.16, that's tests for extraneous agents in viral vaccines for human use. It addressed, so these two chapters, they address the testing of cell substrates and the testing of viral seedlots and viral harvest. They foresee the use of broad molecular methods such as NGS. So as an alternative to the conventional in vivo test for extraneous agents, as an alternative to NAT for specific viruses, or in addition, or as an alternative to in vitro cell culture tests. In addition, chapter 5.2.14, so that's on the concept of substitution of in vivo methods. Which provides guidance on how to substitute, so how to replace in vivo methods where head-to-head comparison is not possible. So that chapter also includes a dedicated example on the substitution of in vivo method for extraneous agents by NGS. And this is, so this example is no longer fully up to date. And I think that was mentioned also yesterday. So there is currently an exercise to update it, to bring it in line with the current thinking. Background to the elaboration of European Pharmacopoeia chapter on NGS. So as you heard, the pharmacopoeia chapters 5.2.3 and 2.6.16, they mention HTS, and they foresee its use as part of this testing strategy for extraneous agents. However, no description of these methods or any guidance for validation is given. The availability of regulatory standards, including validation guidelines in the pharmacopoeia, would serve as a reference for regulators and manufacturers, while HTS was recently introduced in the revised ICH-Q5A, and you heard about that in the previous presentation. And while a panel of model viruses developed by the FDA was also recently adopted as WHO International Reference Panel. And I think you'll hear about that also more in the coming presentations. So in this context, a new European Pharmacopoeia chapter on NGS has been elaborated. So the document is entitled High Throughput Sequencing for the Detection of Viral Extraneous Agents. So that's chapter 2.6.41. So that's a draft general chapter. So it contains a description of HTS technology with the different steps of the HTS workflow and important consideration for each of these steps. And then it provides guidelines for validation of HTS method. And the idea also for this document is to be complementary to the revised ICH Q5A guideline. It was elaborated by the European Pharmacopoeia's HTS Working Party, which is an international group of regulators, national control labs, and industry from different regions, so Europe, US, and Canada. It is still a draft chapter, and it underwent recently public consultation in our public forum, Farm Europa. The public consultation ended on end of June for the public and end of August for the national pharmacopoeia authorities. And the stakeholder comments are being examined by the group of experts. So all the elements that we are going to present in the coming slides should be regarded should be considered in this context, so that this is still work in progress. Quick overview of the draft chapter 2641. So it's a draft chapter describing the methodology, HTS methodologies. So used for the detection of viral extraneous agents. So in biological products, such as vaccines, proteins, viral vectors used in gene therapy or cell-based preparations. So it outlines the different steps of the workflow, design of the method, so non-targeted or targeted, analysis approaches, so genomics, viromics, transcriptomics, and also the controls that are used in the routine test. So it gives guidance for method validation, and that includes recommendation on the selection of the spiking material for validation, then the evaluation of the relevant performance characteristics for HTS, so as part of the generic method validation, and then the product-specific validation, so for the particular matrix. Considerations on the replacement of in vivo and in vitro tests by HTS are already provided by the revised SEH guideline Q5A, as you've heard in the previous presentation. So these are not repeated in the chapter 2.641, and instead we've included a cross-reference to the revised ICH Q5A guideline. So this slide shows a high-level structure of the draft chapter. So with part 1 defines the scope of the chapter. Part 2 is about describe the method with the different steps of the workflow, sample pretreatment, extraction of nucleic acids, post-nucleic acid extraction treatment, library prep, sequencing, bioinformatics analysis, scientific evaluation of the results, and follow-up investigation. And this section also includes a dedicated part on the controls in the routine assay. Then part 3 addresses HTS method validation with some general considerations. Then the spiking material for validation. And then the generic method validation and product-specific validation. And finally, part 4 discusses targeted HTS. So introduction and scope. So to ensure product quality and safety, a comprehensive strategy is established. So following the principles of viral safety risk assessment that are given in the Pharmacopoeia chapter 517. HTS can be introduced in the viral extraneous agent testing package so to fill gaps that were identified in the risk assessment, to replace the conventional in vivo test for extraneous agents, to replace NAT, or to replace or supplement the in vitro cell culture test. HTS is especially useful for the testing of new cell lines, so to detect known and unknown viruses, but also of virus seeds and harvest, and particularly when there is interference in conventional tests. So as you're aware, there are different HTS technologies, short read and long read sequencing technologies, and also different approaches. So a non-targeted approach, so to detect a broad spectrum of viral extraneous agents, known and unknown. So using a comprehensive database with viral sequence diversity. And then a targeted approach to detect a range of known viruses or unknown viruses related to known viruses, using selective capture or amplification of viral sequences, or using reference virus genomes for bioinformatics analysis. So now moving on to the part on a description of method, general considerations. The selection of the most appropriate HTS approach in a given product should take into account the potential contaminating viruses and the sample type, which may be cellular or cell-free to be analyzed, with consideration given to the detection of cell-associated and cell-free viruses. So different approaches exist for sample preparation, depending on the test material for detection. So either of all genomic viral nucleic acids, so that's genomics, or viral RNAs for transcriptomics, or encapsulated viral genomes for viromics. And the workflow that is applied should have sufficient sensitivity in the relevant sample matrix for the potential level of contamination at the stage of manufacturing. And this is to be shown during validation. So the draft chapter also includes an illustrative figure. So the figure shows examples of HTS workflow. So depending on the nature and source of viral nucleic acids to be detected, depending on the type of material, then a suitable workflow is selected. Some of the steps are optional, so they are marked as such in the figure. And I also should highlight that this figure is going to evolve, also to take on board comments that we receive in consultation. Sample pretreatment may increase the sensitivity of HTS by concentrating viral particles or by reducing background cellular nucleic acids. And the chapter gives some examples of sample pretreatment, nucleus digestion, freeze-thaw, or centrifugation. Extraction of nucleic acids, so that may be performed by isolating, purifying RNA and DNA simultaneously to obtain total nucleic acids, or separately to obtain total DNA or total RNA. And there are different types of extraction methods, and the chapter gives the example of silica, magnetic beads, or precipitation-based techniques that may be used based on the sample material. Post-nucleic acid extraction treatment. An enrichment step may be applied to reduce host cell nucleic acid content and thus increase the method sensitivity and facilitate detection of low-level viral contamination. The chapter mentions ribosomal RNA depletion that allows highly abundant ribosomal RNA species to be removed, and also degenerative oligonucleotides to capture and remove host cell DNA. Library prep ensures that the library generated and the resulting data are suitable for the sequencing approach and representatives of the targeted nucleic acid.
Gwenaël. So I will continue with the sequencing. So as you have seen, we have several platforms. We have seen that yesterday, the diversity of the technologies which are available. So of course, you need to select the most appropriate sequencing technology and platform depending on the intended use. And it should take into account the sample type. Is it high level of contamination or low level of contamination, which is expected? What is the required sequencing depth and the coverage and sequencing accuracy and read length? Of course, generating more reads in a sequencing run may increase the probability of detection of the presence of extraneous viruses. A particular point that we also discussed yesterday, the prevention of contamination. If multiplexing is performed, care should be taken to avoid these cross-contamination, which are always possible. And an appropriate internal control is added to the sample prior to loading the sample into the sequencer. Here it's an overview of the bioinformatics analysis. I will not give more details because the figure is subject to change as well based on the comments we have received, and it will be long to explain it, but clearly you need to have a bioinformatics analysis for a virus detection involves building a pipeline that generally includes initial processing of the input raw reads to obtain quality reads. In some cases, it will be followed by de novo assembly to generate contigs and from overlapping reads, and prior to mapping or aligning the reads and contig using a reference virus or database of viral sequences. So this is a general overview of the workflow, but again, it will be slightly modified. An important step, it's the scientific evaluation of the results. So the validity of the HTS run should be based on the recovery of one or more internal control or on expected results from a control sample run in parallel. Also, the interpretation of the results obtained by the bioinformatics analysis should take into account the predefined criteria for distinguishing true positives and false positive signals. It is recommended to report the positive results at the viral species level. You may also have to document the non-viral and background sequences that can occur. And again, appropriate controls have to be in place to capture any cross-contamination from the facilities or instruments. Follow-up investigation. As you know, we may detect some signals, and it doesn't mean that there is a problem, but you need to conduct follow-up investigation. So when a positive signal has been identified, you have to conduct a laboratory-based follow-up, which is potentially after a first evaluation of your bioinformatics results, of course. You have to interpret the bioinformatics first and assess whether the viral sequence is associated with an infectious virus or not. And you have 3 questions here, which are fully aligned with the questions that you will find in the WHO document, which was published several years ago. The 3 famous questions are the nucleic acids that were found associated with full-length intact genomes. Are the nucleic acids particle associated? And are the particle infectious in cell culture? So for that purpose, to answer to these 3 questions, you have a toolbox of various methods that can be used. It doesn't mean it's an exhaustive list. It's an example of approaches, but you may use long-range or overlapping PCR, sequencing with long-read technology, for example, other methods to answer to the second question, such as transmission electron microscopy, mass spectrometry, PCR, And of course, the infectivity assay, you have a list of methods that can be used to answer to the question. E.g. transcriptomics with additional HTS or viral replication by qPCR, or of course, for retroviruses, for example, product-enhanced reverse transcriptase assay. So really use the toolbox as a list of examples depending on the problem that you are observing with the signal. Of course, we need a number of controls, and I will not go into details because this will be slightly updated as well in the revised chapter based on the comments we have received. But clearly, you have to have a control of the whole workflow in order to ensure that the performance of the test is adequate. An external or internal positive control is included in each test run. And you may have also a number of other controls to test the library preparation step or the sequencing step. One important part of this new general chapter is the validation guidelines, with general consideration for this method validation, with particularly the fact that the validation should demonstrate the suitability of the method for the intended purpose based upon the sample type, because you may use it for cell substrate, cell banks, virus seeds, harvest, unprocessed bulk, and the testing approach as well, with what we have described before, genomics, viromics, or transcriptomics. It is clearly a qualitative limit test, and as such, specificity, including breadth of virus detection and limit of detection, are the parameters to be assessed according to ICHQ2. And as you have seen, we have followed the principle of ICHQ2, but we are not totally aligned with ICHQ2 regarding, for example, the number of replicates and so on. So it's based on the principles of ICHQ2. The validation requires an end-to-end assay validation and may include subdivision of the workflow into modules. And this is what we call the modular approach in the chapter, corresponding to the different steps of the workflow, such as sample and library preparation, sequencing instrument, and bioinformatics analysis. What does it mean? It means that you may want to validate each step individually in the modular approach. This modular approach has one main benefit, is that it provides more flexibility if you have some changes in your different steps, and it makes that it may reduce the effort required for when you update an individual module, meaning changing, for example, your sequencing equipment. The selection of the spiking material for validation is a very important aspect. It should be relevant for the intended purpose and the chosen detection strategy. Of course, the use of model viruses as spike is relevant for the viromics and genomic approaches, whereas the use of infected cells is more appropriate for the transcriptomic approach. But for genomic and viromics approach in testing cell substrates, virus seeds, and harvest, the reference standard should be viruses representing a certain viral diversity in terms of structure, nucleic acid type, genome size, virus morphology, chemical resistance. They should be highly characterized for genome copy numbers, infectious titer, viral genome sequences. And the WHO International Panel for Adventitious Virus Detection in Biological Products by HTS provides this diversity of virus family and is recommended as the minimum panel of model viruses for validation. For transcriptomics, which is the approach in testing cell substrates, cell banks, a suitable spiking material can be cell infected with different viruses. These model infected cells should be characterized for infected versus non-infected cell counts, with the confirmation of the viral expression, viral sequences, and background host sequences. So, short description of the generic method validation. So you have to document limit of detection and specificity. So the limit of detection is defined as the sensitivity in which viral contaminants will be detectable by the method. The sample should be spiked with a known amount of material to mimic different levels of potential virus contamination and should be added before any treatment of the sample. And the HTS method should be applied end-to-end. A minimum of 3 independent replicates of the end-to-end method is required to demonstrate an appropriate assay LOD, unlike otherwise justified. The LOD should be reported at the level where all spiked-in viral viruses are detectable in all replicates to indicate the worst-case detection limit, and be reported at genome copy per milliliter for harvest or virus seeds, or genome copies per cells for the genomics approach for cell banks, and or by the amount of transcript per cell or ratio of infected cells and non-infected cells for the transcriptomic approach. The specificity is really the demonstration of the breadth of detection for different types of viruses, as well as the correctness of the identification. The identity of the spike viruses has to be as it was expected. And again, the revalidation may be necessary when part of the method is modified and can be facilitated by the modular approach that I described just before. The second part of the validation is really the product-specific validation regarding the specific matrix that you want to test. And here, the comparison of the matrix used during the generic validation versus the product-specific matrix is needed to evaluate the potential impact on the LOD and the specificity. In this case, at least one end-to-end validation run is required to verify that changes to the matrix do not impact the LOD or specificity. Last but not least, the targeted HTS. There is a specific chapter in the general chapter describing the targeted HTS, which is a viral detection method using HTS in which nucleic acids are of known and closely related viruses are enriched prior to sequencing. So it's based on amplicon-based sequencing or hybridization capture, which use a complementary probe to capture these targeted sequences. And the use of HTS, targeted HTS, should be based on a risk evaluation, as the enrichment test techniques rely on the availability of known virus sequences. In addition, the design of the enrichment oligos or probes should ensure that a broad specificity is maintained. There is an alternative approach to what is described at the beginning. It's to have a bioinformatic analysis, as described in section 2.7, that can be modified for targeted analysis using specific virus sequence or reference genomes to have a targeted approach, which is based, in fact, on the bioinformatics part. The validation is quite similar, but of course, you need to demonstrate the sensitivity for viral detection by using the relevant viruses that you want to detect. And the spiking materials, it's the same thing. They have to be highly characterized, as it is the case for the non-targeted HTS approach validation. So as a conclusion and next steps for this chapter, so since 2017, you have seen that HTS has been successfully introduced in European Pharmacopoeia for vaccines and gene therapy viral vectors. Yeah, I think I want to repeat that because I think when I said it, but the chapter 523 and 2616, they are cross-referenced in the text applicable to gene therapy vectors. And I think it's important because sometimes you are a little bit lost in the European Pharmacopoeia chapters, but when they are cross-referenced, they are applicable, meaning that these texts are applicable to 616 and 523 to the gene therapy vectors. As you have seen, also the concept of substitution in the European Pharmacopoeia chapter 5 to 14 can be applied to the replacement of in vivo tests for the detection of extraneous agent by HTS without a head-to-head comparison, to be aligned with the ICHQ5A as well. It's the principle of this chapter. It's to facilitate the introduction of in vitro tests to substitute in vivo test without head-to-head comparison. As mentioned by Gwenaëlle, we are in the process of updating this chapter 5 to 14 to make it clearer. we are also in the process of updating the chapter 2616 and 523 as well to make it even more clear in a way and to be also more aligned with the ICHQ5A aspects. The future European Pharmacopoeia General Chapter 2641 on NGS HTS will provide a detailed description of the technology together with these validation guidelines, which I think are very helpful as well for implementation purpose to support users implementing this new technology. And the European Pharmacopoeia HTS Working Party is examining the stakeholder comments received during the public consultation. So stay tuned. I think we hope to publish the chapter in 2025. And I would like to thank the HTS Working Party members. Most of them are in the room, in fact, and Dr. Johannes Blumel for his chair role in this HTS Working Party, as well as Dr. Thuy Bourgeois, who is online looking at us. Thank you very much for your attention. Just a special introduction. I think it's a great pleasure to invite my co-chair, Dr. Arifa Khan from FDA, to join us on stage to present her perspectives. So as you know, Arifa is the supervisory microbiologist at the Office of Vaccine Research and Review at CBER, US FDA. She's also the co-chair the chair of the AVDTIG, the leader of the AVDTIG since October 2012. She has been engaged also as the FDA deputy topic lead in the implementation working group of the ICHQ5A, also in the European Pharmacopoeia chapter as well, very engaged. And I would like to thank her for her engagement because I think where we are today is mainly due to her engagement. And you can look at her bio on the ebook. You can see that she has published more than 100 publications, and many of them are on adventitious agents, retroviruses, NGS as well. So I think we can really applaud her.
Thank you for that very kind introduction, Laurent. And I should just add that I think the advances and the work that has been going on with NGS, it's been pretty much, I think we've worked together on this with Laurent as well, throughout all the years as well as with many of you. So as I said earlier, it is a joint effort that we are where we are today. So anyway, so I'm going to be presenting on the FDA's efforts, experience, and perspective. So I figured it would be better to broaden this with giving some of the background that has led to where we are in the FDA, particularly in the Office of Vaccines, to present to you the overall perspective. And then I will also indicate where there are some specific I guess, status of NGS in the different centers as well as in other offices. Okay, so disclaimer, of course, is my formal communication, represents my own best judgment, and whatever I say doesn't obligate the FDA. Okay, so I sort of thought that it would be useful to present the outline of the talk in this format. It's an NGS journey. Basically, I could also change the title to my lab's personal journey in CBER OVRR in the Office of Vaccines. And this covers our I guess, our investigative research into advanced virus detection technologies and how we moved to focus on NGS and how that research helped towards the implementation, and now we're at the acceptance stage. So basically, the NGS interest started, very early on. However, the impetus to focus and drive this effort really came when this, in 1997, during the pandemic, as a response to the influenza pandemic, and this was really because in order to address the urgent demand for this coming pandemic, there was a plethora of novel cell substrates that our office was challenged with, and this was as well as new manufacturing platforms for the development of rapid, large-scale new vaccines to address the influenza pandemic. However, we had started seeing this trickle in even before this, and this was really the introduction of new cell substrates for AIDS vaccines. There had to be designer cells that can, provide the susceptibility for developing AIDS vaccines, as well as some for viral vectors that were used for vaccine development. So this just sort of pushed the whole situation very quickly when we were dealing with all these cell substrates, so many years ago. And then it was recognized that there was a need for broad virus detection to address not only potential known but unknown viruses in these novel cell substrates. And so there was a lot of discussions in the Vaccine Advisory Committee, which is the public and discussion format in our Office of Vaccines for addressing challenges and getting a consensus feedback from the experts that are on this committee. And so these discussions actually were initiated in 1998 on the introduction of novel cell substrates. And the last challenging substrate was discussed in 2012. And I'll show you a quick slide about what I'm talking about, what cell substrates. And then, of course, this sort of triggered my own NGS research efforts in-house as well as external in the Advanced Virus Detection Working Group, and the need to establish this community effort to really address the challenges and the gaps in this technology. And before we even focused on this, my lab had already compared NGS with the current other advanced nucleic acid-based technologies at this time, and which included virus microarrays as well as long-range PCR mass spec, which was coined in the Plex ID format for anyone who remembers that. So we did compare, and we found that NGS was the technology that could detect a broader range of viruses without the need to know the sequence of a virus, which the other technologies, which were really PCR-based, short range or long range, needed. So that was the origin of our focus on NGS, and the work in my lab started, I think it was earlier than 2010, but this is all the documentation I could come up with quickly, and it's still continuing. So, the initial work we did was with the Roche 454 system, no longer available. The data is still there and it's still useful, and we still look at, analyze it. There's a lot of data we had. And then moving to the Illumina platform, and then moving to the third generation of these platforms, the PacBio and the Nanopore long-read technology. Yeah. Okay, so so while we were, sort of moving along with this, along came the COVID pandemic, and so there was a more urgent need, for industry also to try to use this technology for large, rapid vaccine development, and this was specifically to try to accelerate the vaccine development by replacing the traditional conventional assays, which are more tedious, as you've heard from the details that Johannes presented. They have limitations, and also they take a minimum of a month. And I'll present a little bit of that also, just to add on to what Johannes has presented already. And so in our office, we started seeing actually NGS submission for a novel cell substrate as early as 2009. And now currently NGS has been used for replacing or supplementing one or all adventitious virus assays. So we've seen the whole gamut here. Gamut of applications, and the bottom line is that NGS is accepted as an alternative method to conventional virus detection assays. And this is where we are now. And I can present some of the details of this general statement, I should say. Okay, so this just indicates the early traditional animal-derived cell substrates that have been used for U.S. licensed vaccines, embryonated eggs, and chicken fibroblast primary cell cultures, the diploid cells, MRC5 or WI38, and until a certain time, as I mentioned, the introduction of all of the different cell substrates, Vero was the only cell line, the only continuous cell line, non-tumorigenic Vero cells, which meant low passage Vero cells, was the only cell line cell substrate that was used, and some of the vaccines are indicated here. I mean, it's still used, but now we have expanded the cell substrate repertoire. And the vaccines at that time were mainly inactivated or live attenuated vaccines. And again, the different types of viral vaccines have also now expanded. Okay, so along came the need or the demand for new product development. I mentioned it was started with AIDS and triggered by the pandemic influenza, and now accelerated with the COVID. But the original introduction was due to the need for designer cells to provide complementary genes for viral vector vaccines. The decision that the limited supply of eggs for influenza vaccines triggered the investigations of new cell lines for large rapid production of flu vaccines. And also there were new expression technologies. For example, again, this was for one example for influenza vaccines is the insect baculovirus expression platform as a new manufacturing platform. And new expression technologies means, using different types of vectors for expression of the product. So, some of the novel cell substrates are listed here. From mammalian, we had both tumorigenic and non-tumorigenic Madden Darby canine kidney cells. We have these different types of human transformed tumorigenic cells, and these do have complementation ability for vectors like adenovirus, for AAV type vectors, because they do provide complementation by adenovirus sequences here. And then along came the challenge with not only transformed cells, which are tumorigenic, maybe high cell numbers, but actual human tumor-derived cells like A549s, HeLAs. I know most of you are familiar with HeLAs, as well as CEMs. So, and also, although nobody thinks about CHO cells being a problem for therapeutics, this is the workhorse cell line for monoclonal antibodies. But for viral vaccines, this was new because vaccines, most of the vaccines, well, majority cannot have the same type of inactivation, the robust viral clearance that therapeutics does. And so for vaccines, this needed special attention because rodent cells, tumorigenic cells, and retroviral particles are naturally produced from these cells. And for avian cells, there was introduction of the EB66 duck embryo stem cell line, insect cell lines I mentioned to you, plants, and even bacteria as cell substrates. Okay, so CBER had discussions on the use of novel cell substrates in the format of these various advisory committee meetings. VRPACs started in 1998, and the different issues that were presented are indicated here. We even had discussion about Vero cells for live attenuated vaccines, because until this time, they were used for inactivated vaccines. It's an African green monkey cell line, non-tumorigenic passage, but had discussions for its use for live vaccines. And then moved on to the discussions for in vitro transform human cells like 293/6 that for defective adenovirus vector vaccines, tumorigenic MDCK cells that have been used for licensed product now for inactivated influenza virus vaccine. And then also discussions about the use of the MDCK cells for live virus influenza vaccines. And the final discussion at the VRPAC, and I have to tell you, every time we brought an issue of novel cell substrates, it was like, well, are we going to discuss this again? But it was a different issue. So we felt it was a need to have this final discussion on cell substrates for tumor-derived vaccines, A549s for adenovirus vectored influenza and HIV vaccines, HeLa for the AAV vectored HIV vaccines, and CEM for inactivated HIV vaccine. And actually, it was in this discussion that that the use of whatever advanced virus detection technologies were out there was supported because of the challenges for known unknown viruses. Okay, so you've already Johannes did a great job presenting all the different conventional assays, the general virus detection assays, the species-specific assays. I just want to add that that in our office, based on the challenges we had with novel cell substrates trying to broaden the detection for unknown viruses, we also had included additional assays, either extended or broader PCR assays, expanded cell culture assays by adding more target cell lines, and chemical induction for detection of latent viruses, endogenous or episomal. Okay, so these assays have generally been effective in demonstrating the safety of vaccines. The limitations, Johannes very nicely have gone through all the assays. The one thing I did want to point out was that these are, they take at least a month. The cell culture assays can be 28 days if everything works right. And the animal-based assays, more than 18 days. And for the molecular assays, for the PCR assays, of course, you have to know the sequences. And then you can end up with a large number of assays if you're trying to replace these assays, of course. And then for the chemical induction, our lab developed and optimized a chemical induction strategy. But we realized that you can activate a latent virus, but you're not going to know what it is if these assays cannot pick it up. So that was like sort of my foray into looking at the advanced technologies to complement this detection for unknown viruses with induction. And so I already mentioned that there are specific challenges for viral vaccines, that these cannot include the rigorous inactivation removal steps that can be used in manufacturing of some of the products like biotech products like biotherapeutics, because you need to retain the vaccine potency. And vaccine virus can also call false positive signals in the biological assays and interfere with that assay. And as I mentioned, and some purification steps may be used for some of the newer vaccines like VLPs, viral-like particles or nanoparticle vaccines, but cannot achieve or cannot demonstrate high virus clearance. Again, you need to retain the virus function, but in the case of some of these, the actual vaccine virus itself or the vaccine itself is close to size with some of the model viruses, especially MVM. So that's a challenge for trying to demonstrate the efficient clearance in the system. Okay, so of course then it's become obvious the need for the advanced detection technologies, because you need to be able to detect known, unknown, and unexpected viruses, and this is all the different types that I indicated here. Not only tumor-inducing viruses, latent viruses, occult and novel viruses, but also there are some cell substrates like avian and insect cells that constitutively produce a reverse transcriptase activity. And this activity can be associated with retroviruses, retroviral particles or retrotransposons. So it needs to be evaluated what is the risk of these particles that are produced from these cells. So we also had in parallel, it started public discussions on the new technologies, and started as early as 2009, where all of the different advanced technologies were presented and the challenges with them, and this is published in the conference proceedings in the PDA journal, and followed by a discussion of the technologies and risk assessment by an IABS meeting in 2011. And then and a lot of the discussions were also held in the PDA meetings to address adventitious virus challenges, and we had meetings in 2011, and all of these are also published. And the meeting here recognized that that there were knowledge gaps and technical gaps for further development and application of these emerging technologies. And then I mentioned about the discussion in the VRPAC in 2012 about the human tumor cell lines and their use, and that was when it was recognized that the use of these technologies for evaluating, or at this time it was more to supplement the traditional assays in these cell lines to expand the evaluation. And then even in WHO in 2013, there was a cell substrate meeting that discussed DNA-based detection of adventitious agents in cell substrates. And then in November 2013, in the FDA meeting, the challenges were identified and priority areas to be addressed were identified. And this is really what or prior to this, initiated the thinking that we need to work together as a consortium and to develop this working group that has been referred to. Okay, so but during all of this time, what really pushed everyone to think about and about using NGS, industry, CROs, regulatory agencies especially, was the unexpected finding of the porcine circovirus in a licensed rotavirus vaccine. This was published in 2010 but they shared the data a little bit earlier with the company as well as with us, and so this really highlighted the use of NGS in biologics for known or novel detection. But so PCV1, you may know, this is a known virus, so it's not something. So this doesn't demonstrate its use as a novel virus. I mean, it's just shows the limitations of the conventional assays. It's just that the 9CFR assays do not pick up PCV. It doesn't cause CPE. It's not picked up by any of the readout assays. So this does show the significance that NGS can pick up even a known unexpected virus, but it was actually the finding of a novel rhabdovirus in the SF9 insect cell line that's used for baculovirus express vaccines that was discovered by Helen Ma in my lab using NGS that we were evaluating the RT activity produced from insect cells and found in the NGS data that this novel virus was there. And this was really challenging because it was very distantly related to any virus that was present in the NCBI databases at that time. It was only a partial sequence of a distantly related virus, and it had very little homology, 300 base pair region, 66% identity. So we had to actually get the whole sequence by walking the genome and putting pieces together. And so this really demonstrated the strength of it for novel virus detection and really, and this is what helped us develop the extended bioinformatics strategy for virus detection. And in both cases, it should be noted that the testing had been done, extensive testing. In fact, degenerate PCR primers had been used for the SF9 cell by the sponsor, as well as they had even also done NGS, but they missed detection of this virus, was it needed more attention in its detection. Okay, so I'm going to now so of course the introduction of the recognition of NGS triggered FDA efforts further, I guess more at a higher level than my lab for application in biologics. So the FDA and CBER established genomic working groups and to create a research regulatory infrastructure to support policy development and decision-making related to application of NGS. There was support to strengthen the in-house laboratory as well as bioinformatic expertise for NGS analysis. And then, of course, we initiated the establishment of the PDA supported advanced virus detection technologies interest group with focused efforts on addressing the challenges for standardization and implementation of AV detection in biologics. And this was the first publication just describing the group, and at some point we'll update it with our current efforts, but we've been trying to show that in the presentations. So just very quickly, this is just the overview of the working group. It was started as a users group, a small users group. of experts that had experience with the technology. And once we identified, the challenges, then we expanded it to others, and it became an interest group in 2014. And then now we've been elevated by the PDA to the status of a working group in 2022 because we have outcomes that their interest groups are not really, I guess, set up to do, but our group has been moving forward very quickly. And so the mission is to advance next generation for viral risk evaluation by providing an informal scientific forum for discussions and scientific collaborations. And the initial focus and the ongoing focus is still NGS. Their co-chairs, myself, as well as Simon Nig from Canada, Ken Kono from Japan, and Nomi Dierner from Belgium. And we have more than 240 participants and more than 60 organizations. It's a very broad participation from different agencies and industry, as well as service providers, academics. And we have meetings by telecon once every 2 months, and there are additional meetings of the different subgroups. So the strategy was to form focus subgroups to address the different challenges that were identified in the last PDA meeting. And this was to focus on sample selection, preparation, processing, virus standards, reference materials, complete, correctly annotated virus reference database development, and bioinformatic pipelines, as well as follow-up strategies to confirm the identity of a hit. And as these groups matured and the work matured, and we found that the A and B could coalesce and have joint efforts, and subgroup C still stands alone for the database, and then D and E have also coalesced to join their efforts. So in CBER, in OVRR, there were efforts towards implementation, particularly from my lab, and these include collaborative efforts to determine breadth and sensitivity of virus detection by short read and long read NGS platforms, and this is with the working group, as well as funding provided through Nimble GHF. And then there's also the development of reference virus stocks that you've heard some about already, but we now have the CBER NGS reference reagents to support NGS development and advancement, provided through the NIID BEI. Catalog number is indicated here. And these were previously established as WHO reference reagents based on collaborative study of 2B that I'll mention to you next. And now we have a new panel this year that's been established by a new collaborative study outside the actual working group, but including members of the working group. And this has provided a broader panel of 7 viruses for adventitious virus detection for their NGS qualification and validation studies. And they are available for distribution. I was a little bit, I guess, surprised to hear that they're not available, I guess, to everyone. They are available from BEI to everyone. And I think the lack of availability may be more due to geographic or regional limitations. And we're very much interested in having further discussions so they can be made broadly available, even if it means to have some stock provided in a different region to make it more accessible, because we do want people to be able to use them. And they are to be used as a panel to demonstrate breadth and sensitivity. And then we've also generated the reference virus database. There's ongoing consultation with the working group. Currently version 29 is available and is freely available for downloading at the University of Delaware website. So I'm only presenting this not to go through all of the different spiking studies from the subgroup AB of this working group, but that have initiated different studies in different backgrounds to evaluate the limit of virus detection using short read technology, including viromics and genomics and transcriptomics, as well as long read NGS, more recent studies, and some of these use the NGS CBER reference viruses, and the more current ones are using the WHO reference panel. And there's one that's specific for transcriptomics here, as well as this one. The red indicates the studies that my lab has been involved in. So, and this has been published. This was an early study that sort of laid the foundation for some of the other studies. And then this one study is papers in progress. And then the other ones are at different stages, and some are being initiated. Okay, so these are the different viruses that are the reference viruses that we've been referring to. The original 5 are indicated here. The full panel are including the human coronavirus as well as the MVM. And these viruses were selected based on various properties in terms of the particle size, in terms of whether envelope or not, genome topology, genome size, as well as physical chemical resistance in terms to help evaluate the extraction process itself. These have different genome properties, double-stranded DNA linear, single-stranded DNA, single-stranded RNA dimeric, single-stranded RNA linear, double-stranded RNA segmented, single-stranded DNA circular, as well as single-stranded RNA, very large size at all different sizes, and the single-stranded DNA linear, here the MVM. And the purpose here is that it really focuses, the genomes are important because that is what the nucleic acid-based technology needs to be challenged with. So these are representative of different virus families in terms of different virus genomes, so they can help to evaluate any novel viruses. They're well characterized for all of these properties: infectious titer particles, genome copies, adventitious virus analysis, host copy number, reference virus genome sequence, variant analysis, and we recognize that 2 of the viruses, the EBV stock contains the squirrel monkey retrovirus, and the PCV contains endogenous porcine virus because of the production. These were viruses that are contaminants in these production cell lines, and we've also characterized these viruses as well as their titers in the stocks. And then stability studies have been completed and ongoing, and each virus is individually vialed to allow freedom for custom mixing as needed by the user. And then in terms of the database, this effort was initiated to address the deficiencies in the public databases. I'm not going to go through details because you're going to hear a talk by Peju Chin later about the database efforts and the enhancements. It provides high diversity, maintained and updated by Peju, and available through the University of Delaware. And there's continued work on the annotation by Peru and Trent together, as well as with the Delaware team. Okay, so the ongoing efforts for NGS implementation is through collaborative studies for increasing sensitivity of virus detection in complex matrices and the development of SOPs and test data sets to facilitate the routine NGS implementation. You will hear some of the details of this in the talk by Alison Armstrong later, and she's going to describe the results of our head-to-head study comparing NGS with in vivo assays and in vivo assays and in vitro assays. And then we also will hear from the working group on spiking study 4 from Valeria later that will give details of their long read data. We have in-house studies ongoing, continuing to evaluate performance of the different NGS platforms for virus detection in cell substrates. We've also focused efforts on development of cell-based standards for testing sensitivity of virus detection in cell substrates and cell therapy products. Please visit the poster by Sandra Fuentes next door. And then I mentioned database enhancement efforts by Peru Shin will be later in the talk as well. The status of NGS applications in the FDA. So in CBER, NGS data is currently under review. It's used for adventitious virus testing of cell banks, virus seeds, as well as bulk harvests. It's used for genetic stability of virus vaccine, as well as cell substrate characterization. CBER has an advanced technology team, CAT, that coordinates the scientific discussions on new technologies, including NGS. And within our office, we highly recommend that sponsors request a technical working group discussion related to the use of NGS for vaccine safety and characterization. This is in the format of a non-regulatory meeting to discuss your plans for use of NGS, because this is a major effort we recognize using NGS. It's expensive. So it's better if you're starting, then request a technical discussion with us. It is not a formal meeting. It's much easier to set up. You can get in touch with me or Robin Levis to do this. And this is really to reach a consensus of what's expected prior to all of initiating the study. And this has been quite successful, actually, and also to communicate what we would like to see in the submission. So we do offer this in our office. And we are actually also working outside our office at providing consultation to the Office of Therapeutics, which is the new office name for Gene Therapy and Cell Therapy Office. And because they're also receiving applications for NGS for adventitious virus testing in gene therapy, as well as other applications, but I'm just focusing on the NGS AV testing. And there's an emerging technologies team in CEDAR that is involved in discussions on NGS also for biotherapeutics. So, so we all work together, consult on trying to align our thinking for the for what is being asked to be submitted. But I should mention, we're working on that. Okay, even though this is within the same agency, we're working on aligning, the expectations, and so you really, if you're ready to submit and discuss, I suggest you really go to that office where you plan to submit and have that discussion with them, and they will, ask for consultation from our office. But it's better to get, to know what their specific thinking is. But like I said, we're all working together to try to reach a consensus. But there are different products, different patient population may have different expectations. And then additionally, very interestingly, the CVM, Center for Veterinary Medicine, is also having discussions with us on NGS for AV detection for animal-based biologics. Okay, so as I mentioned, so there is general acceptance of NGS for adventitious virus detection for the replacement assay, in vivo assays, PCR assays, as well as supplementary or replacement assays for the in vitro, for cell substrate characterization, especially when there are concerns for occult and novel viruses, and also when there may be assay interference due to lack of effective neutralization of the vaccine virus, or if there could be toxicity due to the sample. And it also can be a readout assay to reduce assay time as needed. But because NGS is broad, you will get a positive signal. So upfront, you should have your follow-up strategy planned out for what to do if you get a positive signal. And NGS can also help you design a custom assay. If you get the signal, you can identify the virus. You can set up a custom infectivity assay, and then that's sensitive for that virus, and then you can look to see what's the relevance of that signal. And this just indicates the various applications for the mitigation strategy as a test strategy. And then these are the examples in our office of the submissions we've received using NGS. As I mentioned, we saw we had a submission as early as 2009 for cell substrate. We've seen additional submissions in which it's been used for characterization of working cell banks, EOPCs, moving to master virus seed bulk harvest. And these are the different type of applications that we've seen. And more recently, to replace the in vivo assay in a licensed product for testing the master virus seed as well as the bulk drug substance. So this just gives you an idea of the range and that we're working with. More recently, in our office, we've got NGS in regulatory submissions and also in Office of Therapeutic products for genomics, transcriptomics, viromics. Short read NGS platforms are the ones we've seen so far. Variety of biological materials are testing, as well as replacing the in vitro or in vivo assays in one or more stages. And this one particularly refers to our office, I should say. I think for the other offices, they're moving more cautiously, learning, getting experience, but they're providing it from our office as well. And for CEM, there's NGS considerations, and then in CEDAR, there are discussions on the use of NGS. Okay, the general experience of NGS in OVRR submissions, use of reference virus, general expectations We expect the use of reference viruses, the WHO reference panel, or an equivalent, and you can actually provide the data or a cross-reference for the material, for the data, and then to use non-targeted NGS bioinformatic analysis for broad virus detection, for replacing the general virus detection assays. We want details of the sample preparation. Don't be afraid that you submit something and we won't understand it. We will. Please, submit everything to us. We would like to see it, or we're going to ask you if there's something missing, then it's a lot of time used. So sample preparation protocols, including sample pretreatments, any synthesis of the library, cDNA, SOPs and results. We do look at the SOPs. We would like to see how the results, to interpret the results, we need the SOPs, and just to confirm, verify, or provide any further comments. And then we need the assay qualification and or validation reports. And here, I think you've already heard the difference between the two. And then the extent of data that is needed may be flexible based on the submission package, and demonstrated by virus spiking studies conducted in product-specific or relevant material. The details of the NGS platform, including total reads, quality reads, number of reads analyzed. It's always nice to see the pictures if you've got mapping data. We love to see the pictures. It's much easier. And then on the method validation report to demonstrate the throughput is sufficient to reach satisfactory detection limit for breadth and sensitivity of virus detection. And then for the details of the bioinformatics approaches, the pipelines used, including your host removal, the subtraction of the expected sequences. We see that sometimes, non-targeted analysis. So whatever you've done in your pipeline, we would like to see. This doesn't say you have to do these, it's whatever you've done. So just examples of just to include everything pretty much. The different data formats, and it's nice if you want to provide Excel sheets for your hits. We would like to review those, as well as to the justification why you're excluding those hits as being real viruses. And the mapping data, like I said, is useful for visualization, and to specify the virus reference database that's used, if it's RVDB or other, the version of the database, even the date if you have it, the source, of course. All those help to understand the final results. And then the details of the tools, program parameters, and the cutoff criteria, please, for why you call something a positive or negative. Additionally, if you have a signal, you've heard about the follow-up strategy, and this is something that that basically we developed in-house based on our experience with the rhabdovirus work, was we had to evaluate to see what's the relevance of that signal. And here for the follow-up of the NGS signal, calling criteria for including, excluding, the verification of the results if you're following up, to sort of address the questions. Can the results be confirmed by PCR and other assay? Is it a complete virus genome? Are there particles present? Are the particles infectious? Is there a replication competent virus? And can the nucleic acid be quantified? So this will all support why you're including or excluding something if you do see a signal. And then the review of the various submissions is being used internally at this time to develop a knowledge base for us and to create a general format as a reference for formally expecting, the information that could be, provided in a guideline later on. And then the implementation for NGS, it's encouraged because, or it's accepted because it does increase the efficiency time, there's ethical reasons, it reduces animal use, and the assay does provide a specificity for LOD, repeatability, and accuracy. And as you've heard, it's a limit assay, so it's a little bit different in terms of what's expected than the Q2 document. And the current cell substrate and viral safety guidelines and regulatory guidances, they already provide the flexibility for using alternative approaches with broad virus detection capabilities and fit for purpose. And I've indicated our US 2010 guideline also indicated that, as to the WHO, the early pharmacopoeia and the recent ones, as well as the more recent, of course, is actually formally in there now in the QR2, Q5A, and the 2.6.41 that's expected next year. I'm not going to read this because this is the same as what you've seen from Johannes's slide, and I think it just further indicates our alignment in the FDA with the Q5A a document in terms of what is expected for validation, qualification, and verification. The definitions are in agreement here. And we agree we don't need a head-to-head comparison of NGS with existing methods. However, as I mentioned, in some cases, there's still some discussion between the different offices and their thinking. And so I think for the in vivo, there's alignment. And then, of course, for the in vitro, it needs to be evaluated based on the package. And then, and here the expectations for the NGS reports. We want the generic method validation report to demonstrate the capacity of NGS for breadth and sensitivity of detection, the product-specific, matrix-specific qualification reports to demonstrate absence of interference for the NGS virus detection. And I've mentioned this, that in some cases, we've seen the generic validation is done in the matrix. So then you're home free. You only need one report. We're not saying you have to repeat it again. But if it's done in a different matrix or not relevant matrix, then we would want the product matrix specific validation. And it depends on your method validation report really as to what's the extent that you need for the product specific validation report, qualification report. And then of course we want the actual study report of the test samples with SOPs and the results, including the controls. And I can't tell you how many times we have to ask for this, the actual study report. I mean, yes, we can have the method validation, but we also need your study report. Okay. So of course, there's still more work for routine implementation. We have continued in-house efforts and external efforts. We are working on optimizing the pre-treatment conditions to increase the sensitivity of virus detection in these projects, as well as developing SOPs and reference data sets for broader NGS implementation, recognize the development of other types of standards may be needed, and also the database enhancements efforts are continuing to increase the accuracy for virus detection and reducing the follow-up of positive signals. And I'm just highlighting that even in the 2010 guidance, we did allow you can use an alternative approach if the approach satisfies the requirements of the applicable statutes and regulations. And, we can try to figure that out, but yes. So it does give you the flexibility of using other methods. And this guidance provides, it's for characterization, qualification of cell substrates, viruses, other biological products, materials. Okay, and thank you for your attention. I'm sure I've gone over time. Okay. So it's my pleasure to introduce our next speaker, Ivana. Be ready, yeah. Niksevic. She is a scientist team lead on the norms and standards for biologicals at the WHO, and we really look forward to her, the WHO perspective on the use of HTS for detection of adventitious agents in biologicals. Thank you, Ivana.
Thank you very much, Arifa, and also many thanks to the organizers for inviting me to share WHO perspective. And one thing that I would probably recommend before I start talking about high throughput sequencing and WHO perspective on it is highly recommended actually to have coffee break after 1.5 hours. Which is standard WHO practice, 10:30 to 11 is usually coffee break. So I feel very sorry for standing between this talk and coffee break, especially for those who are online following this and those who are supposed to have dinner instead of coffee break. But I'll try to entertain you with everything that we are doing in WHO in a shortest possible time slot. So for those who are familiar with the work of WHO, the first couple of slides will be very familiar, but for those who have not been involved in WHO activities, I'll mention a couple of basic issues regarding the WHO standards. And I'm very grateful to colleagues who presented before me because WHO initiatives have been mentioned several times. So I guess that you are at least becoming familiar through this morning discussion about that. I'll also mention the Expert Committee on Biological Standardization. I'm secretary of the committee, and there are lots of initiatives that were actually done by this committee, and in particular in promoting innovation and scientific developments and advances into practice. Now, standards for high throughput sequencing is the main topic of my presentation, and then I'll share feedback from regulators on HTS for detection of adventitious agents. And you already heard from several speakers this morning what is the situation in some countries, and I'll show you a bit more. And then a couple of WHO events will be also shared just for information with no details. When we talk about WHO norms and standards for biologicals, what is really specific and I would say unique is that WHO, as a UN agency responsible for health and well-being of WHO member states, it's important to mention that WHO has 194 member states. And you can imagine how difficult it is to meet expectations from different parts of the world, different regions, different countries when it comes to health and the actual priorities. So on the link that is provided at the top of the slide, you can find a lot of details that I won't be able to elaborate on, but I'll just mention at this point that when we talk about written standards, these are basically recommendations and guidelines published in so-called technical report series. These are well known as Blue Books, WHO Blue Books, technical report series that were mentioned, and both documents that are relevant for our discussion today and tomorrow were published in that format. In the middle of the slide, you can also see that we have a number of documents that apply to all biologicals, those that apply to all vaccines, 58 documents that apply to specific types of vaccines, which for those who are familiar with these documents, you know that we are starting a bit earlier than documents issued by other bodies. So the most frequent question that I received in my almost quarter of century career at WHO is what is the difference between WHO, ICH, EMA, FDA documents, and you can list the rest. So to avoid that question today, I'll just simply say that the unique role of WHO is to provide detailed guiding principles, a lot more details that you will find in ICH, EMA, and other documents. And this is based on the need in 194 member states, where in many national regulatory authorities, and I just want to emphasize that the main purpose of these documents is basically to serve as a basis for establishing national regulatory requirements in 194 member states. So that requires thorough information about the production and then quality assessment, non-clinical and clinical evaluation. So this is really a very broad scope that we are covering. And that's why in some of these documents, you can actually find the details and specifications that may not be mentioned in other documents. And I'll talk more about the concept of WHO guidelines in a minute. I just want to mention here that measurement standards that many of you have been using, and if you didn't, it's highly recommended to use the panel that was mentioned many times this morning. These measurement standards are essential elements for development, licensing, and lot release. For those who are familiar with vaccine lot release, you know what that means. The most important issue is the development and generation of scientific evidence, and you are all aware how many challenges we are facing when defining the first set of guiding principles for any of those issues that I mentioned. The same challenge is for measurement standards. The scientific evidence is actually constantly evolving, and you can actually see a perfect example of that application in the case of these panel of 7 viruses that started as 5 viruses. And today we have to ask ourselves, what is the next? Are we going to add more viruses? When, how, and why? So the concept of WHO guidelines, I just want to mention that basically we have to leave the space to regulatory authorities to define additional principles, because our documents are advisory, as WHO is advisory organization in its nature. So Providing the principles for setting national requirements means that national regulatory authorities have full right to add more, so that they may be actually more stringent, they may be more specific, they may need to be more detailed in any aspect of these documents. But we would like to see that at least all countries are using WHO documents as a basis. And it's not just a basis for setting national regulatory requirements for evaluation of biological products, but it's also the basis for WHO pre-qualification that also many of you may be familiar with. This is a tool and service that has been provided for a long time to assist countries that do not have expertise, experience, nor capacity to conduct entire evaluation of biological products. It's also important to highlight that these are living documents that need to be further developed, and that's why we have at this point in time a long list of requirements for revision of these documents, because they usually live for about 10 years or so. When Laurent mentioned a few years ago, that was actually 11 years ago. And we need to actually update these documents, because without these updates, we are actually receiving complaints that these out-of-date documents may be actually serving as barriers to the access to up-to-date vaccines and other biologicals. We also provide assistance for the implementation of the guidelines, but I'd like to mention that the responsibility for implementing guiding principles into regulatory practice is with national regulatory authorities. The responsibility for implementation into manufacturing practice is with manufacturers. So WHO role is basically to facilitate these processes, and our implementation workshops are typically designed to have regulators, manufacturers, academia, other experts, sometimes other health professionals around the same table, so that people can actually understand each other much better than when they are dealing officially with submission and evaluation and typical regulatory process. So in WHO workshops, we do offer an opportunity for clarifying why certain principles are as they are and how to apply them to specific cases. So examples and case studies are provided for that purpose. And they're organized at global, regional, and as national workshops involving regulators. Several of these workshops were also mentioned this morning. Trainings, as I mentioned yesterday, this is something that WHO countries are always asking for, and in many cases, we've been able to provide this service. So the question today is also, who is in a good position to provide this service, and is this something that we can maybe work on in a kind of networking or kind of exploring who is in the best position to take this forward. Now, the last point is actually very important for our discussion. This is considerations of the guidance issued by other bodies. So when developing WHO documents, we really intend to complement the existing documents rather than to create a conflict. Actually, avoiding a conflict with other documents is the most important as a basis for regulatory convergence, where again, we would like to see all regulators being on the same page, being transparent and clear so that manufacturers know exactly when to come to relevant NRAs and what data they should submit. A lot of work conducted by WHO collaborating centers and custodian laboratories for biological standardization actually summarized in this slide. I won't be able to actually explain who is doing what, but in essence, there are 8 collaborating centers. The lead center for measurement standards is an institution known as NIBSC, which is now MHRA in UK. And collaborative studies that probably many of you participated in were led by colleagues from MHRA with participation from many institutions, national control laboratories or other industry laboratories or academia participated in these collaborative studies. All collaborating centers are contributing to the development of written standards, and we are trying now to involve all of them in this discussion about high throughput sequencing, and I'll show you where we are at the moment. Collaborating centers have very well-defined work plan, and they are going through the process called redesignation every 4 years. So CBER completed redesignation at the beginning of this year. In the autumn, we completed redesignation of Health Canada. MHRA is the next one coming in July next year. And we also have custodian laboratories, meaning that these laboratories are dealing with distribution of the standards. So MHRA, Polarlich, CBER, and EDQM. EDQM is specifically responsible for antibiotics. So great work done by collaborating centers and custodian laboratories. The documents that were mentioned this morning, I'm just basically showing in 2010, and Laurent mentioned yesterday, that in fact discussion started in May 2007 in WHO working group on cell substrates and cell banks. And it was a lovely sunny day in Geneva with a view to the lake. We were surrounded with beautiful flowers and a very difficult question: how to deal with detection of adventitious agents. People are complaining about in vivo assays. Everybody wants to move out of that. Everybody was ready to introduce something new, but we didn't know what to offer at that time. So imagine 2007, May, discussion during the coffee break. That's why we love coffee breaks, because all innovative ideas are coming out with the smell of coffee. And we were actually, at that time, we agreed that we should encourage development of this methodology. So I just extracted the text that you can see in the TRS 978 from 2010, when we are basically saying, yes, it should be developed. Now, this is the question that you will see at the end of my presentation. We are asking, should we update this document, provide an addendum, or maybe update another document about risk assessment, or maybe develop something completely new that will be entirely focused on HDS. The next step was also defining what exactly we wanted to see as part of the practice. So in the same document, we elaborated a little bit more. We included references that were available at that time. You can see now how much it is out of date. And at that time, it was clearly a research tool, which we were all hoping to see one day as a regulatory tool. And that's where we are today, to see, is this really regulatory tool, or it is regulatory tool maybe in few regulatory authorities, but we would like to see this much broader. The next document that was mentioned, Scientific Principles for Regulatory Risk Evaluation on Finding an Adventitious Agent in a Marketed Vaccine. This is actually the document from 2013. That's the one that was actually mentioned earlier. So all these details that Laurent and Arifa provided, and also Johannes, about the finding adventitious agent in already licensed vaccine, risk assessment and all, that document is also out of date. So we should either update this to bring scientific advances into it, or maybe provide something separate that will be much more focused on this issue. Now, we are flying from 2013 to 2024, skipping all the rest because it was already covered by previous speakers. So I don't need to go into cell lines that Arifa elaborated, but that was all part of the WHO consultations conducted in the past 15 years. In March this year, we actually had the 79th meeting of the Expert Committee on Biological Standardization. You have here access to the executive summary, access to the full report that was published, and also links to the website where you can find all these details. But the important issue is actually extracted here for you on the left-hand side to show this panel of viruses. And people are saying now, oh, this is new. It was just established in March. Actually, it's not that new. It was new in 2020 when everybody was actually busy and obsessed with the COVID pandemic. And we didn't have a proper introduction of that panel of 5 viruses, which in WHO plan, in the original plan, we actually wanted to follow up this establishment with the implementation workshop. But of course, the world has changed in the direction that nobody really wanted. So we are now coming back to this idea, thinking about the implementation workshop, maybe beginning of 2026. We'll see what is the best timing in collaboration with other partners and many collaborators that we are working with. But you can find here the details that were mentioned. So this WHO panel, we would like to see how it works in practice. Because people, if everything works well, nobody is coming back to WHO to say it works well. If something doesn't work, or if people are facing a problem, then we hear about that. People are saying, oh, you know, the panel the WHO established is not good because we cannot actually get this and that. So we haven't got such a report. Maybe this will come first to ARIFA's working group. We don't know. But anyway, we are prepared for implementation workshop because it's really important to go into practical details of how it works, how it's used. Are people really using this as it is intended to be used? Now, details from the WHO document that you can find, 2 additional viruses, and that's why we are saying because 5 viruses have already been in place and provided, I think we should probably go into deeper analysis with ARIFA and other colleagues to see what was the actual feedback on this first panel and how this new panel is going to fly now in coming years. But the most important, it is available. ARIFA mentioned the problem with distribution. The problem is also that sometimes people are asking, is it free of charge? Yes, it is free of charge when it's requested by regulatory authorities or national control laboratories. It's not free of charge for manufacturers. Usually, WHO standards, usually they are charged. But the problem is actually that just paying, for instance, the cost of transport is a huge deal for many WHO countries. So it may not be a big problem for, you know, big pharma in Europe or in US to pay the cost of shipment, but shipment cost is a big issue. And in many cases, WHO is not able to support that, although we are trying to find solutions and these issues. So in this case, as ARIFA said, it's free of charge. We are trying to increase distribution all over the world, and we are looking for potential options so that we are not relying just on one center being distributor, but maybe having more. Now, I'm going to share with you feedback from regulators on the HTS status. In August this year, we actually asked the questions to regulators to explore and to understand what is the current situation. Is this the tool that regulators are prepared to accept and use? And what they actually need. We were trying to identify the areas where technical support would be requested, and also what are the expectations in terms of the existing WHO documents. So 16 regulators responded. They are here mentioned as part of different regions. Many of them are WHO collaborating centers, but I'll show you what you'll see what each NRA is doing or is asking for. So the first question was, does your country accept HTS as an alternative to existing in vitro and in vitro adventitious virus detection methods for viral safety test of biological products in marketing authorization and or clinical trials? So 75%, which is 12 NRAs, responded yes, and you can see them on the slide. No was actually from India, Japan, Korea, and South Africa. And countries have already adopted HTS as an alternative or in process of adopting, discussing its adoption. So it's work in progress in many places, but at least you will see where NRAs are at the moment. So most countries are ready to accept this as alternative for both in vitro and in vivo assays. China, alternative for in vivo assay only, and Ghana, alternative for in vitro assay only. From Tunisia, we didn't get details, but we will probably receive this. What are the challenges for not accepting HTS? So we were then interested for those who said no, why not? So India responded a lack of WHO documents with request for mandatory HTS testing for adventitious agents. So that just shows, and we expect that this would probably be the answer from many other countries, because when we are encouraging development of certain tools, it's not seen as a requirement or recommendation. It's just seen as encouragement. So maybe that's not enough, and that's one of the reasons why we are thinking when and how to update WHO documents. Another reason in India is that there are no validated methods for HTS. When we were discussing with colleagues in Japan, they said lack of experience and or performance in a country. The reality is actually that at least this is what we understood, that is the currently Japanese manufacturers are not submitting HTS data to regulators. But we believe that this data may actually soon come to regulators all over the world. So that's why it's a question, what should be the next step? By the way, regulators from who I'm mentioning in this feedback, most of them, I think, are virtually connected. So if I say anything wrong or if I misinterpret, because it's a sensitive issue, what is the view of regulators from different countries, we'll have a chance to correct this in the panel discussion. And I invited all of them to participate in the panel. Koreans were very detailed in saying that there is a lack of regulations or guidelines. A few years ago, the response was, we are waiting for ICH guidelines. Now ICH is in place. There are also details that lack of experience or performance in country, NGS technology and criteria for assessing the validity of HTS-based methods have not yet been fully standardized. For cell and gene therapy products, validating HTS methods is challenging due to limitations such as interference from genomic nucleic acids. And also HTS data considered supplementary. So they are not accepted as alternative to the existing assays. In South Africa, regulators are saying that HTS has not been adopted as policy. This is still under discussion. The question 3, if you answered yes to question 1, which products has your country authorized for the use of HTS for viral safety test? So for vaccines only, it's actually China. USA, and in Indonesia, it's a very specific situation. I think Indonesian regulator is online as well. The HTS has been used for characterization of poliovirus type 2, NOPV2, as many of you know of, but not for detection of adventitious agents. And they are not yet there for the replacement of in vivo or in vitro assays. But at least the tool is there for characterization of vaccine, which I think is a good sign. Tunisia is accepting this for vaccines and biotherapeutics. Germany for vaccines and cell and gene therapy products. For biotherapeutics, there are no applications so far. This is what Johannes clarified for us. And then for vaccines, biotherapeutics, cell and gene therapy products, we have Australia, Bangladesh, Brazil, although they don't have approval so far, Canada, Egypt, and UK. And in Ghana, no application so far. In response to the question 4, in which steps of production manufacturing does your country accept the use of HTS for viral safety testing? We got different responses, as you can see, Bangladesh and India for cell lines and other raw materials, Japan and Indonesia for appropriate steps in the process of the production, Australia, Brazil, China, Ghana, and Germany for cell lines and raw materials, as well as for the production steps. And Canada, Egypt, Tunisia, UK, and USA for all, from cell lines to the final products. We didn't get reply from Korea and South Africa, so this is still pending. The rationale for use of HTS, question 5, by which rationale your country accept HTS, and question 6, if you selected A or B, please specify relevant regulations. So we got a different response from different countries, WHO and ICH, Pharmacopoeia, submitted method validation report of HTS by manufacturer. So countries are having different options, and I leave these details. The presentation will be available, so you can actually dive into that. But it's basically something that if we broaden the survey will be even more different from what we see from this small sample of 16 NRAs. Consideration of WHO recommendations for HTS. So when we were basically mentioning that the acceptance or suitability of HTS for adventitious virus detection in the evaluation of biological products, did you consider WHO recommendations? We got yes from 7 participants of this brief investigation, and no from 8. So WHO documents are not always considered, as you can see. And question 8 is, if you answered yes to question 7, please provide your feedback regarding the need for updating or elaborating more on adventitious virus detection. So 5 countries responded that WHO documents need to be updated or elaborated: China, Egypt, India, Indonesia, and USA. And there are different reasons to include detailed description of the method, the new method appendix with a high quality reference viral genome database. This is the database expectation that I mentioned yesterday at the beginning of the workshop. Egypt wants to have a comparative table describing the pros and cons of each method, indicating relevant and feasible methods for each manufacturing step and for different types of raw materials. They also want to see description description of the state-of-art methods that should be used. India would like to see a revision to align ICH and WHO document with ICH document to develop sequence database and bioinformatic tool. Indonesia would like updated guidelines on viral safety test using HTS. And from USA, we got response that update of WHO document to align HTS with other current ones, with ICH and also with EDQM chapter. So we're all looking forward to seeing this updated chapter from EDQM next year, and maybe to start WHO consultation on this in parallel to see what is coming next. The international reference panel, we were just asking, are you aware of that? We didn't even dare to ask, are you using this? We were just asking, are you aware of that? And surprisingly enough, we got 3 responses that people in regulators in Australia, Indonesia, and Tunisia are not even aware of that. The others at least know that they exist, and we repeated again using the questionnaire as an educational tool also to introduce this, so that those who didn't know should become aware of that. And just to mention a couple of WHO events in this and next year, due to the very short time slot, I did not really go into regular updates on WHO initiatives in different areas that the audience might be interested for. But here is just to mention that the expert committee meeting on biological standardization that was held in October, the executive summary is available on the website. The detailed report is in preparation and will be published at the beginning of next year. There was a 19th International Conference of Drug Regulatory Authorities held in October in New Delhi in India, so-called IGDRA, or as we call it friendly, Olympic Games for regulators, when lots of issues have been discussed and one of the topics, for instance, post-approval changes for vaccines and other biologicals, also reduction, replacement of animal tests used in the evaluation of biologicals, cell and gene therapy products, regulatory framework for these products, and many other topics. And these recommendations from IGDRA are also available on WHO website. Two events at the beginning of next year. In the last week of February, there will be WHO drafting group meeting to discuss revision of recommendations for the preparation, characterization, and establishment of international and other biological reference standards, 24 to 26 of February in Geneva, WHO. And another one will be informal consultation on WHO guidelines. Actually, these guidelines are on the phasing out animal tests in the quality control of biological products, 27 and 28 of February in WHO Geneva next year. There will be also implementation workshop on cell and gene therapy products in April next year. That will be the second one in the African region. The first one was held in Eastern Mediterranean region in May this year. And the second one will focus on the needs of African countries, and implementation workshop on the evaluation of biosimilars in July next year. And then the next expert committee meeting in October 2025 will be 13 to 17 October, 81st meeting of the expert committee, when we are going to discuss new standards and issues submitted to committee for review. And I really want to express special thanks to colleagues who provided feedback to the questionnaire on HTS. Everybody listed on this slide should be actually part of the panel, or at least most of them. Several colleagues are here in the present in person, and the others virtually connected. So at 2 o'clock this afternoon, we'll discuss this, but really this is a great feedback from countries. And also many thanks to WHO colleagues, members of drafting and working groups who are conducting a lot of this work, colleagues from collaborating centers and custodian laboratories, and many individual experts and stakeholders for support. And also very special thanks to my colleague Yoon Kyung Kim, who is working as a secondment in WHO and is really helping a lot to move forward in this area. So thank you very much for your attention.
And thank you for the organizers for allowing me to speak today. I will explain what that unwieldy acronym means, but I think what I really want to impress upon you or want you to take away from this talk is that through the look of historical data that we've collected for viral contamination events, as well as in vivo and in vitro data, I want to convince you that not only should you not be afraid of NGS, but you should be afraid not to use it. All right, now this. Okay, so viral contamination events have a very large impact. First and foremost, they have a big impact on the patient, obviously for safety reasons. But, you know, luckily, almost all of the time, patients don't actually, even when there is a contamination event, that doesn't necessarily reach the patient, but there's still a big impact because, you know, there's downtime that has to take place in order to deal with the manufacturing costs, and investigation and CAPAs that have to be put in place, and that can lead to product stock out, which could be an issue for patients that are waiting for life-saving medicines. As I said, there's a manufacturing impact, there's a regulatory impact as well. Companies may now be exposed to intense regulatory scrutiny if these contamination events occur. And there's also obviously business impacts as well, especially if it becomes public information. And so because of that, many companies don't really disclose their viral contamination events. There's no obligation really to disclose them. And a lot of the times they're motivated by concerns for negative publicity. And so when they do disclose these events, which happens rarely, they rarely describe this event in any sufficient detail to be of significant value. And they're really only able at this point to learn from their own contamination events. So because of that, we at the Center for Biomedical Innovation at MIT worked together back in 2011 to set up this consortium called the CAACB, or Consortium on Adventitious Agent Contamination in Biomanufacturing. I will not say that again, to fill this information gap. And so we launched this back in 2011. The goals of the CAACB were as follows. So we wanted to use this as an industry consortia where we could share experiences in the control and risk mitigation of adventitious agent contamination by manufacturing, provide a forum for networking, identifying best practices, conducting collaborative research activities, which I'll talk with you today about, as well as promote generation and application of new technologies such as NGS. We do this through a number of methods. First and foremost, we host 2 workshops a year. These are interactive closed-door meetings just with our members that allows, because they're closed door, it allows our members to freely discuss case studies and have open discussion around viral safety. And we also have a number of member-driven projects, and I'll talk about 2 of these today that are perhaps of relevant for this meeting. Before I get into that, I just quickly want to thank our CAACB members. I know there's a lot of you here that are present in the room. We have a number of CROs, CDMOs, drug sponsors, suppliers that all participate in this, and we really appreciate your support. So the first project that I want to talk to you about was our or I guess I would call this like our flagship project. This is a living project where we collect and analyze viral contamination data in biomanufacturing. And the premise of this was, as I said, collective intelligence is more valuable than individual experience. And the confidential collection of industry-wide viral contamination data and subsequent risk analysis assessment would be a highly valuable lessons learned exercise for industry and could guide companies in best practices to mitigate the risks that lead to these events. To do this, we first set out a scope for what we wanted to cover. So just to be clear, we only focus on mammalian cell culture, rDNA biopharmaceuticals, and cell culture produced vaccines, and anything at the manufacturing scale. So we don't look at anything that's in discovery or QC. We don't look at vet products or fermentation. We also only focus on viruses, and all of the data that we collect is only from our CAACB members, which include drug sponsors, CMOs. We look at GMP and non-GMP manufacturing and anything collected since 1980. The way that we set about this project was we gave a 166 comprehensive survey to each of our members, asking them to detail any contamination events that have occurred, any false positives they've had from tests, controls that were in place at time of contamination, the extent and source of that contamination, any determination of where the process was breached, what the containment and investigation strategies were, the decontamination strategies, CAPAs put in place, cost of the event, and any risk management strategies afterwards. We collect all this data, we anonymize this data, and then share it amongst our members. And we've published a paper on this back in 2020. But this allows us to kind of collect information that normally wouldn't be easily disclosed. So at the moment, the CAACB is aware of 29 viral contamination events that have been reported either to us or publicly. The ones that are highlighted in bold here are the ones that have been reported directly to us, and those are the ones that will be involved in the study today. Yes, it looks like that most of these events happen in either North America or Europe, but that's just simply, that's where the manufacturing has taken place from our members. That certainly doesn't suggest that other geographies would not be affected by contamination events. So I want to share 3 insights from this survey. The first is that viral contamination events, as you expect, are rare. So we've only had 21 of these reported to us over 35 years based on volume. But they're not rare when you look at it on a per company basis, and they're quite expensive when they do occur. So getting more into the numbers a little bit. So we had 23 companies complete this survey. 10 companies out of those 23, so 43%, reported a viral contamination. But when it comes to the number of events, we had 21 total events. So you can do the math here and see that companies definitely experience sometimes more than one contamination event over the lifetime of the manufacturing that we asked for. Companies also reported 16 false positive events, or there were 16 companies that reported at least one false positive event, with 31 events reported in total. And as I said, these can be very expensive. So looking at the investigation alone, every time an event occurs, this can cost at least $1 million to upwards of $50 million to deal with this investigation. That doesn't include the cost for discarding materials, decontamination, and corrective actions that have to be put in place afterwards. And we weren't able to collect any data on other ways that things can be expensive, such as, if things become public, a decrease in company stock value, loss of revenue from a product stock out, et cetera. So this is why we're all here, right? We want to develop a strong viral safety strategy to either prevent contamination events or at least detect them early on in a process to avoid this. When contamination events do occur, the mean manufacturing downtime was approximately 1 to 2 months, but we had some people report upwards of 3 or more months while they investigated and dealt with this contamination event. Okay. So the second insight that I want to share with you, and I think this is important when we think about NGS a little bit, is that CHO cell cultures, which is the primary workhorse cell line used for monoclonal antibodies, were contaminated by different viruses from different sources compared to human and primate cells. And the reason I want to highlight this insight is because when you are developing an NGS assay and you get these complicated bioinformatic results, it's important for you to understand your process. You want to understand, is this a realistic virus that could be in my process if you get a positive hit? Because that can help inform how you go about investigating that moving forward. So looking at a little more detail here, you can see that the majority of the events that were reported to us involved the CHO cell line. That's not necessarily mean that CHO is more susceptible. It just means that there was a higher volume of CHO being used. But in the viruses that are in solid here at the bottom of this legend, the majority of which are MVM, you can see that they are all different from the viruses that infected the human or primate cell lines on the right. If you look at this in a little more detail, you can see that this table here shows that the viruses that are in black now, these are the ones that infected the CHO cell lines, mainly came from media components, animal-derived serum. This is how that entered this process. But the viruses in red were mainly attributed to operators. Okay, so that makes a lot of sense. Operators are human. They can, sometimes get infected with a virus that could be then infect your human or primate cell line. Okay, so maybe most relevant to this topic here. The other insight I want to share with you is that virus safety testing does have clear limitations, but when it's used in a targeted way, it can prevent virus spread in a facility. Let's look, for example, at quality control lot release tests. For all the events that were reported to us, in 11 of those, or I should say, in almost all of those events, an in vitro virus test was used for QC lot release at the time of the event. There is an immunofluorescence test here at the bottom. That's basically an endpoint measurement of an in vitro virus test. And you can see that in most cases, it did detect the virus that was involved in the event. But there were actually 5 cases where the in vitro virus test was unable to detect the virus that was eventually implicated as being the source of the contamination. Okay, that's actually a pretty high number. So that suggests that, having this in vitro virus test as your only QC lot release test may not be sufficient to really detect what could be out there. We had one event that reported a qC-PCR test, which did detect the virus. Looking at a little more detail about how these viruses were initially detected, confirmed, and identified, in most cases, they weren't actually initially detected by any sort of assay. They were mainly initially detected because there was a change in the cell culture performance. So the cells started to die, and then it was like, okay, what's going on here? We gotta figure this out. There was one case where NGS was used to do this initial detection, but in most cases, as I said, the other assays that were put in place didn't do that. From a confirmation identification standpoint, though, molecular-based assays by far were, well, I shouldn't say by far, but were mainly used for this purpose. So you can see that PCR confirmed the virus in 10 of these cases and identified it in 11 cases, while NGS was able to identify the virus in 7 cases. Now you can see some other technologies here. I should note that this is historical data, right? So this is data collected all the way from 1980. So some of these technologies have, are not in favor anymore, and NGS didn't exist as a technology for this until, the mid 2000s at the earliest. And so over time, it's pretty clear that these molecular-based assays are really critical for confirming and identifying a potential event or the virus in a contamination event. When companies went back to look at the source, though, and they tried to identify, okay, where did this virus enter my process? Let me look at the raw materials. In most cases, it was inconclusive. They were unable to use assays that they had in place to determine for sure that that virus came from a media component. We only had 3 events that reported a direct detection and identification of a virus from a media component, even though it was suspected to come from there. And in all of those cases, they used a molecular-based assay like PCR. In one of those cases, they even had to concentrate the raw materials in order to do the detection. Now, as we've heard in earlier talks, PCR or NGS is now considered as sensitive as PCR. And as we know, PCR is a targeted approach. So if you don't have a PCR, if you have an unknown virus or if you have a new virus that affects your event, your PCR is not necessarily going to be able to detect it. And so you can imagine nowadays with all the talks that you're about to hear, if you could replace even your PCR with NGS or have it as a side-by-side assay, you now have this breadth of detection that you could use to help with this issue. Not only was molecular-based assays useful for the determining the source of the contamination, but it also helped prevent spread to downstream. Okay, so out of the 21 total events, 7 of those events spread to the downstream process. And in all of those cases, virus tests were not implemented as forward processing. Now, that's partially likely due to the fact that most of the compendial assays simply take a very long time to run. And so, people move forward, they didn't realize that there was an issue until it was too late. In the cases where it was contained to the cell culture, in some of the cases, this was just due to the cell death, or the material was stockpiled upstream for testing. But in 3 of those cases, there was virus testing that prevented forward processing prevented downstream contamination. And in all of 3 of those cases, it was a PCR test that was able to detect the virus and prevent that downstream or prevent the process moving forward. And so once again, this impresses upon you the fact that if you have an NGS test that is as sensitive as PCR, but more agnostic, it could be extremely useful to avoid a spread to the downstream, which is much more costly and difficult to deal with. We also asked companies or members what new technologies were applied during these events. And you can see that in the majority of cases, PCR was a new technology. That means it wasn't in-house at the time of the event, but it was developed to try to understand what was going on during the investigation. And in 6 of the cases, NGS was developed as well. When it came to contaminant-specific assays, 43% of the events developed a PCR assay as a kappa for detecting this virus in the future. Although many companies didn't develop any new assays. But as I said, some of this is historical And so I think this would change if you look at more recent years. But if we asked them what the most useful tool was for viral contaminant identification, and now you can look at this on a year-by-year basis, you can see that NGS now was considered the most useful tool for identifying the virus when the contamination occurred. You can see starting in 2005, NGS pops up on this graph right around the time at which this was being considered. And now in the more recent events, it outweighs even PCR as being the most useful tool. Okay, so the key takeaways from this project: virus contamination is extremely low. There are different viruses that contaminate human and primate cells typically than CHO cells. And then while cell culture performance was the most frequent initial indication of a contamination in 2 events, performance did not change. The in vitro virus test is the gold standard, or was the gold standard, for broad spectrum viral contaminant detection, but it's lengthy. And in 5 of the events, that assay did not detect the contaminating virus. Rapid in-process virus detection methods have been used to contain the contamination in bioreactors. And during the investigation, testing of raw materials was positive in only 3 of 11 events. And those positive results required some molecular-based assay. And what I didn't mention here is that large-scale downstream viral clearance was effective at removing most of these viral contaminants, but it's not really pertinent for this discussion today. Okay, and you can read more about this data here in this publication from Paul Barone in 2020, although I did update the data here because this is a living project, and so we're continuously getting new data as this goes along. So some of the numbers you would see in this paper are not the same as the numbers I just presented with you here. Okay, the other project that I want to talk to you today is the historical evaluation of the in vivo virus test and its potential for replacement with NGS. So at the time that we took this survey a couple of years ago, CAACB members continue to report the use of the in vivo test. 94% of them use it for cell line characterization, and about half of them use it for virus seed testing and lot release. And since 2000, our members have performed more than 10,000 in vivo tests using more than 84,000 animals and more than 67,000 in vitro tests. So we asked the question, what is the value that the in vivo test is really providing here? As you saw in the earlier slides, I didn't even mention the in vivo test when it came to actually investigating a real contamination and so forth and so on. So this was a paper, others have mentioned it, that came out back in 2014 that did a head-to-head comparison between the two. Generally speaking, in vitro performed better than in vivo, although there were a couple cases where that wasn't true. But there really was no public record of historical in vivo testing data to understand if these sort of head-to-head results hold up in practice. And so the premise of this work was to collect historical data from our members. So we collected data from 20 of them on their experience with the in vivo test, the in vitro test, and the use of high throughput sequencing for viral safety. And so we asked them, over the lifetime of their use of the in vivo test, have they ever experienced a positive result on an in vivo test that was not detected on another supporting assay? The answer, no. There's never been an in vivo test that was the only test that detected that virus. There was always an in vitro test or a molecular-based assay or something else that supported that. Okay, so it does really beg the question, what is the value of this assay? What are we running this for? Why are we doing this costly, lengthy assay that also involves the use of animals? Let's look at the data in a little more detail here. So there were over 10,000 in vivo tests done and over 67,000 in vitro tests. The false positive rate for the in vivo test was around 0.2% or 21 total events, while the in vitro test was a magnitude less. And when we look closely at the false positives on the in vivo test, this is just a distribution of those different types of in vivo tests. So you can see a lot of them came in the eggs and mice, as well as 9 of them in the HapMap portion of the HapMap rat test. And when these false positive events occur, they take a lot of time to resolve, right? So you have to now investigate, when you have an in vivo positive test, what happened here. And so on average, this can take 1 to 3 months for you to have to investigate something that isn't necessarily true, right? And in one case, we actually had someone report that it took over a year to figure out that their in vivo test was a false positive. The false negative rate is about 0.03% for the in vivo test and even lower for the in vitro test. We asked specifically, you know, what was the repeat rate for the in vivo test, and in all the cases of the false positive result, that was redone. And as I said, that can take a lot of time. So repeat assays alone can take, you know, and 37% reported it took less than 1 month, 63% reported it took 1 to 3 months. So our conclusion is that alternatives are needed for this in vivo test, and that's sort of why we're all here. As I said, it hasn't been reported to detect a viral contaminant. More than 84,000 animals have been used by members for in vivo testing. And there are also biological products for which in vivo, in vitro testing will not necessarily work, like viral products with no neutralizing antibody. So sequencing is definitely a potential alternative for viral detection. I'm not going to belabor these studies. I think almost every speaker mentions them at one point. There was the PCV1 contamination in 2010. And also, I think Arifa's group also detected the novel rhabdovirus in an insect cell expression system. And I won't belabor this either. You just heard from this. The regulators also agree that you know, NGS is encouraged as a replacement. So we asked our member companies if they are currently exploring or using NGS in their process. You can see that actually very few are currently using it as a validated assay. So we only had 3 companies that said they're using it, 10 are exploring it, and 3 said that we'll look at it in the future. And the way that they want to use these assays are different. So 69% of our respondents said that they're exploring replacing the in vivo with NGS. 50% are exploring supplementing the in vivo with the NGS. So not necessarily replacing it altogether. And as we know, some companies have included NGS in their IND filings. What is the reason behind this replacement? So most companies report that they want to reduce animal use, and they have greater breadth of detection and faster detection. 40% said greater sensitivity, and 20% said less expensive. I think we had one company that said it was a company directive, which is nice to see, actually. I wish more companies came from the top. But there are some challenges to implementing NGS, but I think that as you'll see in this conference today, those are being addressed. So sample isolation and handling can be somewhat difficult, but the AppTwig group has ongoing work and published work to create industry consensus on this effort, and the new European Pharmacopoeia chapter details this. Bioinformatics is quite difficult. It requires in-house expertise, and there's a lack of guidance necessarily on setting the thresholds for these algorithms. But there are a lot of CROs that are out there now that are filling this need for many clients. So I don't think this would necessarily be a barrier for drug sponsors. At one point, there was a lack of standards, but as you see, there's now a WHO reference standard available. But even despite this, you know, NGS may not work for some product modalities like autologous cell therapies, where analysis time may be too long. That being said, any of those compendial assays would also be too long for those product modalities as well. So overall, I want to impress upon you that we feel that NGS is a critical tool for viral safety control. But developing an NGS assay does take time. You can see how complicated these assays are just from hearing all these talks. And so I want to impress upon you that I think it's important to start now, because it'll save you critical time, you know, should an event occur. You know, you don't want to be one of those people that are now developing NGS after you realize there's an issue. You want to do this now so you really understand your process, understand how you can use NGS with your process effectively. NGS gives you broad spectrum detection in an agnostic manner. It is comparable or has better sensitivity than both compendial assays. It gives you both simultaneous detection and identification of a contaminant. So that's the one advantage over PCR, or that and its broad spectrum detection. And it takes less time than the in vitro and in vivo test. And you can read more about this project in a paper we published last year. And so with that, I want to thank you for your time. I really want to thank Stacy Springs, who's the executive director of CBI, where I work at MIT. And I really want to give a special shout out to Paul Barone, who's the CAACB director. He really should be the person up here giving this talk, but he can't be here for medical reasons. And then Jim Leong and Michael Wiebe for being the lead investigators on this work. With that, I want to thank you for your time, and I'm happy to answer questions during the panel discussion.
Okay, so good morning, everyone. So my name is Noémie Deneyer. So I'm working at GSK, but today I will present the EFPIA, so the European Federation of Pharmaceutical Industries and Association, because we would like to present the EFPIA perspective on the validation and implementation of next generation sequencing for virus safety testing of biological products. So I'm here today, but I'm not the leader of that initiative, but unfortunately they were not able to join this meeting, so I'm, let's say, presenting on behalf of them. So for the presentation, I will first explain the initiative, so behind this EFPIA working group. Then I will present you the position paper that we released, and here, you have the link. So this is a position paper that has been submitted in June this year on the EFPIA website. So it's an industry position paper. And I will focus today on two topics from that paper. So the validation and the comparability study sections of that position paper. And at the end, I will also finalize this presentation with a case study and remaining question from industry that we took from that position paper and that exercise. So first, a short introduction about this working group. So the project has begun to facilitate the implementation of the NGS in relation with the cell line characterization and the viral safety for biological products. And so the first instance was really to have an industry position paper and a way to share, leverage different industry expertise, experiences in the way to introduce and implement the NGS as an alternative for standard viral safety testing. So the scope, of course, is next generation sequencing. That was the main scope. We wanted to have applicable for viral safety of biotechnology products, so vaccines, also recombinant protein. ATMP were out of scope. We base ourselves on the regulatory landscape, and you will see that we worked through that journey through the revision of the ICHQ5A and also the general chapter of the European Pharmacopoeia. So we try to be linked with those updates to be sure that this position paper is adding details on those guidelines. It's adding industrial position relating to those guideline changes. So we were 16 companies around the table, and so we delivered the position paper after a few years of discussion and best practice sharing. So this is the journey. So you see that we began in 2019, still a long time ago, but as I said, we try also to adapt based on the draft of the ICH, the European Pharmacopoeia, to be sure to be relevant on our position as industries. So we started in 2019, then we shared that initiative at the ABDTIG and CACCB as well. And we began with drafting introduction, and the main part was the validation part. So it took more or less 2 years to have this section drafted because we wanted, as I said, to be aligned with the changes of authority. So we share that section with a working group from ICHQ5, but also from DQM. Then we draft the comparability part, and you learned already this morning, but also yesterday for those who were there, the things for the head-to-head comparison, that this wording and what there is behind this wording. So we took that into consideration during that part, and you will see that later. Then we finalized with a regulatory part with some example on how you can submit this kind of NGS technology and recommendation to address the regulatory part. So finally, it has been published in June, already presented for some section in different PDA forum as well as CACC. So the position paper is organized in 4 different sections. The first one is the introduction. So we are in detail covering what is the next generation sequencing. We are comparing that technology also with the PCR, with in vivo, in vitro, other commonly viral safety tests. Then we have a huge validation section, including all the different parts on how you can address the limit of detection, the sensitivity, the robustness, and so on, the different different control that you can use, all about the bioinformatical analysis as well. Then a section on comparability, so how to address that if you want to replace or supplement current assays, and finally, as I said, the regulatory. So today we will focus on the validation and the comparability. So for the validation, and I think I'm quite repeating what has been said by Johannes already this morning, and also by Laurent and Gwenaël. So this method is considered as a limit test. So for the control in impurity. So we need to assess the specificity, the detection limit, and also the robustness. Inside or outside the validation, but we'll come that after. So for the specificity, if we take the definition of the specificity, we need to be sure that the test is not interfering with that you can analyze your analyte without interfering with your matrix. And so for NGS, you can do that with different manner. So for example, you can use positive and negative controls. So for the positive control, what you can do is you can use your sample, you can spike known viruses in your sample, and then you can assess the specificity if your method is identifying exclusively the sequences of the spiked virus in your sample. This is a way to address this point. Also, you can use negative control. And so for the negative control here, you can have a native sample, which is, for example, your matrices without your contaminant or your viral seed of interest, etc. And then you can assess whether it's specific and whether you can't find any other contaminant in your sample. It's not easy. We discussed that yesterday, that having a negative control is not the easy part. Could be included if possible, but we know also the limitation of that approach. For the detection limit, the detection limit is the lowest amount of an analyte that in a sample which can be detected, but not necessarily quantified. So for NGS, the detection limit can be demonstrated, for example, using, again, spiked sample. So you can spike your sample with different viruses, the viruses model, and you have been heard, you heard about those models quite a lot already in the different talk, to mimic different level of contaminant. And then you can then address the limit of detection here. So for example, you spike at 4 different concentration. You can have signal detected only for 3 of them. So you set your detection limit here. Maybe the most difficult part with those model viruses is how you are reporting those detection limits. Because we know that the detection limit will vary according to the viruses, small one, large one, the way it can be extracted and so on. So all those steps are quite critical to define this detection limit. So we propose in the position paper 3 ways to report those. There are pros and cons for all of those ways. But for example, you can assess the worst case scenario. So on your 5 or 7 model, you can say, okay, this is the worst case detection limit, but it's an underestimation of the detection limit of the methods. You can say list a detection limit which is specific for each viruses, but then you are only targeting the viruses you put in your sample, which is not really the thing behind using those models, because they were selected for different type of family properties, and normally they should be extended to all other viruses. Or you can have a range of detection limits between the worst and the best case. Regarding the things that can be spiked, so the model that can be spiked in your sample, you can use either viral particles or infected cells, depending on your test material. For example, for the viral particle approach, you can use it mainly for seed, harvest. You can also use it for cell lines, but for cell lines, one can also recommend to use infected cell approach. I will not enter into the details, but this is a figure from the paper, so you can see the advantage and the limitation of each of the application, or each of the possibility. For the detection limit, as I said, the sample preparation can also have a strong impact on the model. So this is why we are recommending to spike the model contaminants from the start, before the extraction. I think it has been said also already, but this is quite important because extraction is one of the major parts also of the sample treatment. Also, the sequencing depths can also impact the detection limit. So if you have more read, of course, you have more chance to look at low-level contamination. So the sequencing depth can be adapted based on the equipment you are selecting to do your sequencing or your multiplexing also approach. If you are multiplexing more, you have less reads per sample, and so you can impact also your detection limits. Finally, the robustness. So as the ICHQ2 and Q14, the robustness can be included as part of the validation or evaluated during the development phase. For that, to be quick, I think it's a risk assessment. We think this risk assessment to be put in place to highlight the different critical steps of the methods. And then, of course, you have to have those critical steps to be varied and be sure that your method is still robust. Critical steps include sample extraction, library preparation, or sequencing. So this is it for the validation part. And for the comparability, it's true that we base ourselves on the update of the ICHQ5A, where we said that we can replace in vitro and in vivo without head-to-head comparison of the methods. And so we start from that and we ask this question, okay, is it really sufficient to do it without head-to-head comparison? What is it behind this wording? Could we not still provide analytical comparability? Is it not needed? So we said that, okay, if you want to supplement or complement a test, validation or qualification, depending on if you want release or characterization test, could be sufficient. But if you really want to replace, we assess the fact that maybe we still need analytical comparability. And so this is what we assess in the section. And to help us, we use a generic methodology to address this need to have an analytical comparability. So this methodology includes a pre-assessment part during which we assessed whether there is really a link between the different methods to be replaced. So we are, and you can find the details in the paper, but we were asking different questions. So is the intended purpose of the methods achieved by measuring the same product quality attribute? Do the two methods use the same readout? And is the test category the same? For example, limit test and so on. So at the end, we did this evaluation comparing NGS with PCR, in vitro, and in vivo assays. And after this pre-assessment, we had two choices. We can say, okay, if an experimental comparability study is justified, we could need to compare the performance of the two different procedures. If not, if scientifically it's not justified, we need to provide a justification for the replacement. And so we detailed how we could do that. So briefly, for the replacement of the in vivo by NGS, this is the position of the paper. So here on the top, you can see the wording from the ICHQ5A. And at the end, we are quite aligned with this ICHQ5A terminology. And in the paper, we said that the analytical comparability is not useful for in vivo for different things, because we can see, and it was discussed by Charlie just before through the Baron 2022 paper, that there is maybe no need to go in that direction, and that the NGS is suitable for the intended purpose, that the specificity and sensitivity will be demonstrated during the validation. So that's okay. And the breadth of detection is also included through the different spiking approach that will be included in the validation. But still, we still had some question about the justification of the detection limits. So can we make use of comparative data already published in the literature? I think yes, and we heard that already, that to justify the fact that you want to replace in vivo by NGS, you can use data already available in the literature. Of course, taking into account that your NGS workflow can have different performance compared to the one that is published, but it's to take into consideration in this justification. And that the suitability need to be justified based on the intended purpose and additional virus detection assay, including the overall control strategy. Because if you want to replace in vivo, you will still have, I guess, some in vitro testing, other testing as well. So you have to check that, to see that as a global control strategy, of course. So this is for in vivo and for the replacement of the cell-based infectivity assay. So again, you can see here the position of the ICH, which recommends to replace without head-to-head comparison. Here we're presenting maybe a more conservative position about the fact that we still think that analytical comparability will be interesting to provide, and I think it was aligned with what I already heard yesterday and this morning, that the absence of head-to-head comparison is not meaning that you don't have to provide a justification data showing that your method is also critical. It's also therefore it's also applicable for the intended use. And this is for us due to the fact that the cell-based infectivity assays have critical role in the current viral safety control strategy. And we can see that in the two publications from the CSCCB that was summarized just before by Charlie. So, of course, we need still to have, so we need to, for us, it could be interesting to have an analytical comparability. Maybe it can be enough if we are integrating that in the validation. So those are points to be taken into consideration. There is also limitation of an analytical comparability through, for example, the use of the model viruses that we are using. But I think that those viruses we're selecting already taking that into consideration. And at the end, the difference in terms of sensitivity and breadth of detection may be accepted as well. So it's true that, and it was a point raised by Johannes this morning, that we know that for all the viruses, maybe the breadth of the sensitivity will not be the same as the in vitro cell-based assay, but this is something also to assess whether it's acceptable or not based on the breadth of detection of the technology itself. So just to end that presentation, so we wanted to also share where industries are with case study. So different industries are interacting with health authorities. As already mentioned, there is a different way to do that. There is a innovative way and also through more regulatory meeting, but there is also possibility to ask for a non-regulatory meeting with certain regulatory authorities to discuss the topic, and it was already mentioned by Arifa this morning. Mainly topics that were discussed by industries with authorities are the details, NGS workflow, the validation approach and the different results generating during this validation, the nature also and the type of the spiking material, so cells versus virus model, which virus, etc. And when conducting also the comparison results between the in vivo or the in vitro assays and the NGS data that were provided. So in the industry, so they receive, of course, feedback. So for the majority of them, the feedback were positive for the replacement of in vivo and in vitro by NGS. Question raised by authority included data analysis, viral genome database use for the NGS analysis, how you validate also the bioinformatical pipeline, what are the criticality of specific step, the justification on the spiked model used, and also justification of the detection limit. So those were, let's say, the main feedback that were received by the different industries. And so we wanted also to highlight that at least one application was submitted and accepted for the replacement of in vivo. And you can see more details in the paper. So at the end, it's true that we were left with maybe some remaining questions, but I think that today and yesterday, and I guess tomorrow, we will have answer to those questions. So you wanted to say, okay, but what about the replacement of the in vitro without analytical comparability? So is it based on the wording on the ICH, how can we do that? And I think this is a question that will be addressed, or that was already addressed today. The justification of the detection limit, how are we handling that, is still an open question for us. How are we forcing the acceptance of the ICH Q58 statement regarding the replacement without head-to-head comparison in Europe and US, but also in other international countries? And I think that for that, it was really a nice presentation for Ivana regarding the WHO. It's already a first answers. And also the perspective of moving from a panel of orthogonal virus safety tests, so with PCR, in vitro, in vivo, I think we can exclude it for now, but do we want to move from that to a unique NGS-based assay, or how are we seeing as industries and authorities the future of the viral safety testing panel? So it was still an open question for everyone. So with that, I would like to thank you for your attention. And here you can find the name of the lead authors with all the different contributors for the different industries involved in this position paper. So thank you for your attention.
So it's time for the panel discussion. So I will invite all the speakers from this morning to come on stage. In addition, I will invite also Dr. Michael Wall from Health Canada. He's a senior CMC reviewer at Health Canada. And we will have online Mrs. Julati Dalan from the National Agency of Drugs and Food Control from Indonesia, Dr. Adir Abousalem, Egyptian Drug Authority from Egypt, EDA, and Ms. Amida Begum from the National Drug Control Laboratory, the Directorate General of Drug Administration in Bangladesh. Yes, I think you are all there. Thank you very much. We see you on the screen. That's perfect. And the idea is to open first the discussion to the audience. I will also be part of the panel, but it's to open it to the audience. If you have any questions related to the talks you have listened this morning, and of course, we'll also ask to our colleagues online to participate just after. Yes, for all the attendees who are online, you can also ask questions on the chat, and we will manage them as well during the panel discussion. Thank you.
So Peru from USA FDA. So I have a question, or maybe ask the comment from all of the participants and also the panelists regarding the NGS validation for different cell lines. So let's say if we have somebody already have the validation report for one cell line, and by any chance, if the production changes to different cell line, will the validation report need to be redone, or we have some sort of the consensus, like if this kind of changes is within the species, for example, the changes from hex cell line to HeLa cell line, we may consider not to redo the validation report. And I would imagine that maybe you change from the human cell line to insect cell line. Definitely, these two cell lines are so different, so you might need to redo the validation. But I'm wondering to what kinds of the labels that we need to have a new validation report to accommodate the requirement. I'm not putting Arifa under the spot, so.
So thank you, Peru. So actually, we've been involved in discussions on this topic. And of course, I guess that's a broad topic for everyone. And just to add another layer of complexity. So I guess Peru mentioned two scenarios. You've got same species, and then you've got different cell lines from that species. And then you've got, different species, fine. Well, the different species, I think, is easier. But then there's another situation in which if you have the same cell line and different strains of the same cell line, do you consider that? So, okay, so from the product development point of view, of course, if you change your cell substrate, it's a new product, generally. So the question is really relevant to the NGS testing in terms of, I guess, the, what would need, if it needs revalidation, if you have this already validated in the same cell substrate or a relevant cell substrate or a different cell substrate, it's DNA equal to DNA equal to DNA. So, but I can tell you, and I'm not able to answer the question. I'm going to leave it to others because we've been having those discussions with the different offices, different centers, and there's still differences in the viewpoint. So we do appreciate hearing from others as well. Want to put up the line?
Yeah, it works. I will have a first reaction from a technical point of view. I think you need to look if the cell concentration is the same or similar, because I think it can affect potentially the background and the limit of detection, of course, of your test. So I suppose there is something to look at that first, for sure. It's just a technical. If it is the same, Arifa, you want to answer?
I told you we're discussions about it.
Yeah, I think it's.
In terms of there are differences in terms of expectations from the different
Yeah.
offices right now. So yeah, I'll just
Also quite hard for me to give a unique answer, but I suppose it probably happens more often when developing ATMPs, where people are changing cell lines or using other things. So this might be relevant, and certainly yes, I was first thinking vaccine, and then as Arifa said, it would be rather a new product, and so for me really would be a case to case. So how much differs the new cell lines or the new cell clones from the other, and what is the clone? How was it established? Is there a lot of much more animal stuff in or less animal stuff in? So is the background equal or not? So for me, we would ask for some justification and case to case justification. Whether they think that the old cell line or the old background or the old data would be still valid or not.
I'll just comment really quickly too, is that, if you have an approved product and you're changing your substrate for production, you have bigger problems to worry about than the NGS. But if you're early in development and you are, working on different cell lines, I think, most data could still be supportive. It's still your same method. You're with internally. Obviously, you would need verification that it still works, but it's very different than if it's approved product than if you're early in development. So.
But if you have internal controls that are spiked in each sample, and the result is satisfactory for this control, what do you think?
So I guess what you're saying is about, testing for interference or something in the NGS, and if another, I guess, cell control doesn't show interference, and would it matter if you're using another cell line of the same species? And I think that's a different question, and that is also very important, rather than if you're testing the cell substrate. Whereas, of course, if you have a new cell bank, you would have to do new testing. So I think, again, it depends on what's in the cell line. Like, you may have some transgene in a certain cell line, whereas in another one you didn't. It really, I think, depends on what's in the cell line, and if there's something that one can imagine could interfere.
I can imagine that the response is not the same if you are using a viromic or a genomic approach, or if you are using a transcriptomic approach, where you could have internal control spiked in each sample to be tested. You can check that the control are covered correctly, and it seems to be okay.
The internal controls, of course, they help a lot. It's rather a question how much you put in from your internal control.
It's not a question of how much, if the criterion is the coverage.
Yes, the question with the validation is rather whether you are willing to claim the same limit of detection of your assay with the new substrate or not.
Yeah, and I think that on a company point of view, it's interesting to think about the validation of the cell base as a platform method, to say, okay, we will have this full validation package for cell sample, whatever the cell is. And I think then if you are changing and if you want to apply that method for a new cell type, you have to put a risk assessment in place, assess what are the difference between your cell, the previous cell lines and the new cell lines. Then, and I think it was mentioned in the regulatory document, do a matrix verification, be sure that it's working, that the limit of detection is reached with these new cell lines. And I think that maybe the trickiest question is the background, because as you say, depending on the cell lines, you will have different background, and it can be, I think, observed during this matrix verification, and you can assess this background of your new cell lines, but without redoing all your validation from scratch, because you already have a platform method.
That's a little bit provocative. Of course, with new methods, we are always very cautious. And what do we if you change it? So, question back: what would you do when with the in vitro assay, if we would do the same? Would we revalidate it, and how much would we revalidate it?
Except that assay is not validated.
It's compendial and it's anyway perfect. Or, okay.
Okay, so I guess you don't have a perfect answer right now, but I think it is food for thought, for more thinking, and maybe it might come up again in the broader discussion in tomorrow's panel if somebody has some additional thoughts about it. Any other questions?
So I guess we heard it more than once. The in vivo, replacing the in vivo by NGS is generally accepted if you have a validated method in place. And you may do some product-specific verification, of course. When people talk about in vivo, some include the map-up, some don't. And my question is, what is the regulatory body view on this? Is it the same? Or are there any other requirements if you have a validated method in order to replace a map-up as well?
I think the general theme was that the NGS can be used to replace the map-up. Yes, so that the way is general open, and probably also the bioinformatic way to look might differ, because with the map-up, if you have a certain number of viruses that you are specifically looking for, and this is an advantage also in the sensitivity. So probably that there might be two ways in interpreting or assessing the NGS data. Once for the specific virus, or in a rather targeted bioinformatics approach, and the other way is using it rather as a broad detection method. But
so that means
I think that the ICH, the way is general open to use NGS also to replace the map-up.
So that means on the bioinformatics tool, you in principle check for these viruses specifically instead of going against all the viruses, right?
Yeah, yes, you can.
In order to replace it.
Yes.
On the bioinformatics.
Because it'll
level.
make more sense. Well, so it depends. It's there in that way. If you look specifically based on your pipeline, the targeted may be a little bit more sensitive. And then you're specifically looking for that list of viruses that you're testing in the 9CFR. No, this is not 9CFR, that you're testing in the map-hap wrap for the antibody production
Right.
assay. Yes, yeah. So even there there is a list of viruses that each of the species has. So you can specifically.
But you don't have any sensitivity data like for the in vivo.
Exactly, right. So that's the advantage.
That's not an issue. Okay.
No, but for the NGS, you would have to show the sensitivity, of course.
For these viruses which are outlined for map-up wrap.
Yeah, yes. If you're using NGS to replace any of the assays, you do need to demonstrate.
Because it's a specific assay, and that makes it different to the general in vivo screening assay.
Right. So for the specific assay, that's what Johannes was saying. The difference, you still have to show sensitivity, but you want to characterize your assay, the performance. So you still have to show what is the limit of detection of your assay. But the difference is that you can use targeted approach for those specific viruses.
Okay, so it would not be sufficient to go with the 7 reference standards. So you would request sensitivity data for these viruses specifically.
Yes.
Even though that is not provided by the map-up anyway.
If you replace it with PCR, what would you do? I mean, you would still.
PCR is a different. That would be the next question.
Yeah,
but
no, I'm already answering. So PCR assays are currently replacing the map-hap wrap test.
Okay, so I
Some-
just take it, so you're requesting some sensitivity data for all these viruses, which are outlined for the MAPAP assay.
Correct. Well,
Okay.
because if you're using the assay as for replacing any other assay, you want to characterize your assay. You want to demonstrate what is the performance of your assay. Otherwise, if you're saying, it will be eventually if it's a compendial assay, that's a different thing.
Okay. That would mean in principle, coming back to the lab, all these viruses which are outlined, that you don't have these viruses, right?
Well, you don't have it for the in vitro either. You don't have it for the in vivo adventitious agents, but so you don't have detection limits for any of the viruses those
true.
assays pick
Yeah.
up.
But you're asking for such data.
Yes,
And how can we demonstrate it?
with the model viruses or any other panel that's equivalent to those viruses, if you think you've got something similarly characterized. But the model viruses are encouraged because then you can compare, the results across different.
So yes, maybe there is some misunderstanding, is that you're saying that your assay has to be validated using the model viruses, and you determine the limit of detection of your assay using those model viruses. And with this limit of detection, you can say that I can replace those other tests, even if they were specifically targeting some viruses. I know that my assay, my NGS assay, at least is able to detect at this amount, and I'm comfortable replacing those other specific tests without specifically looking at those specific viruses, because the viruses may not be available to assess them within the NGS. And that's the purpose of the model viruses, is that they will represent the overall viral world that we are expecting to look for. And because we cannot compare head-to-head each assay that we want to replace by NGS. And that's the purpose of the model viruses. So I think the misunderstanding was maybe there, and I think we may be aligned.
I fully agree with
You're welcome.
I fully agree with your point. I was going to say the same thing.
Hi, Christopher Bravery. I'm chair the Cell and Gene Committee for IABS. And I'm not a molecular biologist, so you'll have to excuse me if you already told me this and somehow I missed it, and that I haven't really thought about it. But this panel, this reference panel, is that agnostic to the nature of the sample that you might be testing? And I'm thinking specifically if we were looking at, say, cell therapy products, allogeneic cell therapy products, would that reference panel still be suitable? Or should we be looking at some slightly different viruses?
So the selection of the viruses are based upon all the different properties that have been shown. So therefore, it's suitable for any species. It's not species specific. It's just virus characteristic specifics, if you will. So it represents the different properties to test the whole workflow. So it can be used in any background.
Amina from PatoQuest. Just maybe to comment on what was said. If we're talking about cell bank testing with other approaches like transcriptomic approach and not a genomic approach, these model viruses would not apply. So are we, and I know there is some work done there to have reference cellular models where we are with that, and what are today, if we are targeting replacement for cell bank characterization of HapMapRap, what is the expectations from the regulatory bodies, considering the reference models would not apply with the transcriptomic approach?
So I can just tell you some of the information that I'm aware of. So people are using the reference viruses to spike cells. Okay, yes, it's not relevant, it's what is available. And so some add the viruses to the cell pellet, then lyse the cells in a scenario that maybe the cells are expressing virus and therefore they're producing virus. So in that way, so It's not, of course, the perfect standard, but I think some studies use that in that purpose. And that's the reason, there are efforts to develop cell-based standards. And I think you may have seen Sandra's poster there. So those are so last year she presented on another cell clone that we had, which has got latent virus, so that can be used for looking at latent virus genomes. And she presented spiking studies. Now we've got two cell lines that have different levels of virus expression. And actually they're being evaluated currently in some ongoing studies to look at their, I guess, suitability for use in spiking studies. And we're looking at it in a complex matrix. And then there are other clones that we have that may be provided, but the limitation of what we have right now is that it only expresses one virus. And of course, that's the challenge. Are you going to have, how many viruses do you want to have, different cell lines? And I can tell you right now, we're not going to develop cell lines for every of the model viruses. So we're not doing that. And I think it's not feasible to have one cell line infected with different viruses. So the cell clones that we have, I mean, you can, but I think then you have to characterize. Yeah, okay. Yeah, so basically the way these will be provided is similar to the vialed viruses, is direct to be used from the vial. You don't amplify, you don't grow, but then you have different expression. So they've been characterized for expression.
I think there's of course a fundamental difference between the transcriptomic approach and the viromic or genomic, because as when selecting the 9 WHO, we were afraid of different capsid structures and different genomes configuration, like double-stranded and so on. But this does not apply to the transcriptomic approach, where you don't have any capsids and where probably the structure of the mRNA, it's a single-stranded RNA molecule, is much more uniform. And that's why I'm always wondering, but we have anyway the afternoon session about standards and validation. Yes, whether here not even for me, synthetic molecules could be some pragmatic approach.
I agree. We can use synthetic RNAs, I will show in the next talk. And we can also derive cell lines, which are transfected with synthetic RNAs expressing open reading frame or subset of open reading frame for different viruses as a model without using naturally infected cell lines.
Hi, Robert Charlebois from Sanofi. I have a question about the WHO reference standards. So those are extremely good for validating sample extraction through to identification. But from the bioinformatic point of view, they're too easy to detect because they are very well characterized viruses. They have really good matches in the database. But the contaminants that we may find may not be that close to reference sequences. And so I think there's a gap, just like there was a gap in terms of transcriptomics, I think there's a gap in terms of validating bioinformatics. Sorry. There could be a false sense of assurance that your assay is working well, whereas you could maybe only detect things that match the database really well.
So I guess our approach has been for that when you're using the non-targeted approach, you can also test the breadth of detection with the database sequences, because you will see under your parameters not only the detection of the model virus, but you should see how far it goes if it picks up, other hosts or other species that have the same virus, because the reference database contains all species. So I think for the detection that you're talking about, the bioinformatics challenge, of course you can have, the like specific sequences but I don't know how that would challenge it either.
I think we talked earlier about the possibility of having synthetic sequences spiked into the data sets. Those could be standardized. We can have, either using something like what Marc Kellois was developing in terms of, or other evolutionary models to create sequences that are like viruses, but not anywhere close to anything real. I think that would be an important addition to the standards that we use, because otherwise a bioinformatic pipeline that would miss natural viruses is not what we want for viral safety.
Yeah, thank you for sharing that. And this is what I meant when I said that we need specific feedback on how it works in hands of every user. And maybe a working group can actually have a look into that and see, because there are certainly limitations as well. But we probably didn't go into details because we have to hear from users in their specific context what they are facing as a difficulty, and then to conduct the analysis. So everyone who is using, feel free to share this, send to Arifa or any of us, and we'll have a look at that.
I think from my perspective, that this is actually my largest concern, especially on the review side, where you could be getting a submission, everything looks okay, but you're not really sure on the bioinformatics side whether or not it's going to pick up all these variants. And I could see a potential of having, I mean, I think the 7-virus panel is like the foundation. You need something like this. And then having, it's something that's been discussed, is the idea that you'd have sequences that you could put into your matrix. It's not in the capsid anymore, so you don't have to worry about the actual extraction. You can have different variants. And then there's the in silico side. You could just test and see if your process. But then at the same time, you know, as a reviewer, when you get this, you need to know to look for those things. That do they have something on the bioinformatics side for looking for variants? But yeah, I think that's maybe one of the weaker points that we recognize what the methods have right now.
Hi, hello. Yeah, very interesting topic, actually. We have coined this problem, relatedness, the relatedness issue at Novo Nordisk. And what we have done is to do it in silico and to try to heavily mutate the WHO panel and look for that. We have also removed the references from the database and tried to see if we can find distantly related viruses, and that have worked quite well. But yeah, we have also tried different things. But I think it's very interesting topic, and I was actually going to propose that we should talk about how to generate in silico data and which algorithms to use, and yeah, to also align on that, because there are different pros and cons to multiple of them. And I would like to hear your insight on that, if you have any.
I think, you know, I mean, if it's just to test the bioinformatics step, then, you know, something like that, you know, could be used. In addition to, you know, having the other live viruses in there. I think that was proposed also yesterday, to have like a combination of the two for the bioinformatics. I guess I'm still struggling to understand how synthetic, you know, sequences can, you know, challenge the bioinformatic pipeline. I guess it's really in terms of trying to create the diversity that you're looking at. And I guess for us, you know, we've been able to do that with the extent, the evolutionary detection of other species from the same virus family, you know, based upon the non-targeted approach. So I think the different approaches, it can be used. But I think to have a standard like that, at least then it can be available maybe, and that, you know, everybody can use the same standard. That really is more, to me, I see as it more important than, you know, people doing their own non-targeted and coming up with the list of what viruses they can pick up eventually, just for the bio.
For me, the challenging with this bioinformatic standard is, of course, that the background of the raw data really depend on the specific sequencing method and the specific background. And certainly we might reflect as what kind of diversity we want to accept, and what kind of 100 or 200 families should be in, and that's great with the in silico. You can think can create a lot of breadth for the detection. For me, the challenging thing is as what would be the suitable matrix, or would it be okay to have a set of fake sequences, or like Mark Elva mentioned yesterday, a kind of hypothetical novel viruses that could be spiked in silico. But how should the raw data look for this novel method that they really fit to your specific method?
You can made virus pop. It's a database of fake viral proteins. You can use. It's a publication of last year. You can use the database. It is based on an evolutionary distance, and you will see a list of fake viruses that are consistent, and you can use freely to test the capability of your pipeline to detect the distant sequences. So the database exists.
While you're getting that, so I guess one of the challenges still would be that if the sequences are so different that they don't exist, then in the real life situation, you don't have that challenge in which you've got, sequences that, in the real analysis that they may be, distantly related, or whatever the percent homology would be with a known virus. So, I mean, yeah, and also I think it's the actual sequence data and the coverage it gives you. So if you're getting specific sequences, I don't know if your real data would have that region covered, if it's a specific region that you're looking at in your pipeline. So I guess it's really just only for the testing, the reproducibility of the pipeline, and it's, I guess, for that purpose. I have to visualize that.
Yeah, to emphasize this, it could go a little bit deeper, for example, by simulating some artificial reads, either from already sequenced matrix, and you add the data inside. For example, we could be able to evaluate if our pipelines is correctly identifying some virus with a huge homopolymer. We can add a level of complexity on the in silico viral genome, and maybe it could be a work from academics lab or either regulatory to provide a data set where we can launch our bioinformatic pipelines and be sure that we tackle all the questions, for example, with a huge diversity of the reference, as it was mentioned before, to have a really a tool to evaluate specifically the bioinformatic pipeline and either to spike in directly a real-life analysis.
So I think this is a great topic, and I think you've given us a lot of food for thought, perhaps, to sort of have further discussions in the working group as an initiative in next year. And we're also looking for those topics. So clearly this is a topic of interest. But I think we want to move on, give the chance in the online to hear some thoughts from them as well.
Okay, so thank you for joining us, Juliati from Indonesia, Hamida from Bangladesh, and Hadir from Egypt, and also Kim from Geneva. I think it's a good opportunity to share what is going on in your places. I briefly presented this morning what we received as a feedback, but let's imagine, for instance, if the manufacturers are coming to Indonesian NRA with HTS data for detection of adventitious agents, what kind of response they can expect, or what would be the expectation from regulatory point of view? Juliati, would you like to start with the Indonesian situation, and then we can maybe fly to Egypt?
Okay. Thank you very much, Ivana, for the question. Actually, frankly speaking, formally, our current regulation in Indonesia has not yet required NGS, as per also for the experiences on the evaluation for evaluating the NGS also limited for us. However, considering the general concept that we are following the ICH as well, that if the manufacturer or applicant come to us with a proposal using the NGS methods for the testing, we could accept the proposal in, of course, the applicants should provide us with the evidences that the method is having a good validity. So, for instance, since we are talking about the viral safety, for instance, viral safety testing, so we if the company come to us with using the NGS method, then they should provide us with the evidence that the NGS method for the viral safety testing should be, have a good specificity, sensitivity, also, also if they have a comparability study with the existing method, that will be also become our consideration. However, currently we only have one experiences for evaluating the NGS. This is for the NOPV2 application. This is a vaccine. And in that particular cases, that this NGS is mainly have an objective for characterization and also to evaluate and monitor the genetic stability and product consistency for this particular NOPV2. Because usually for the polio, detection of the mutation, we are using MAPREC. However, since the consideration of the practicality of the use of the MAPREC, and also the other limitation of the use of the MAPREC, so that in NOPV2 in the development, the NGS is recommended. That's why for the NOPV2 we are NGS is using for the characterization. However, this NGS also considered for the evaluation for detecting of other polio virus they may be present in the vaccines. So this is why in this particular application, NGS is used by the applicant as a supporting a method for the viral safety, in addition to the the tissue culture that they use for the in official method for the viral safety testing.
Thank you very much, Giuliatti. It's good to hear about this experience with polio vaccine, although we are focusing here on the detection of adventitious agents, but at least having experience in one area that is relatively close to this, probably it's opening the door for further discussion on that. Hadir, what is the situation in Egypt?
Hello, greeting from Egypt. Could you hear me?
Yes, very well.
Okay. First, I want to thank you for inviting me to participate in this paramount important conference. Actually, we concerned with the evaluation of quality module for biologics, and you know that adventitious agent is crucial because it affects product safety directly. Actually, in EDA, we stuck to international guideline for evaluation of A part mainly. And so we depend on ICHQ5A, and you know that the update version encourages the use of NGS for detection of adventitious agents. And we highly appreciate the use of this advanced method by applicants. However, the applicants have to submit documents that support the efficiency of methods for detecting a broad range of viruses. In Egypt, we will request the just a comparability. It's not important to be head-to-head, but at least comparative batch analysis when the applicant shift from old conventional method to the NGS method. Addition to the full method validation report, clear SOP and clear result. That's just to ensure that method is efficient and the applicant able to make good interpretations of the results. As you mentioned that this method is a little bit complex and need multidisciplinary experts to get the correct conclusion. However, unfortunately, we didn't receive an NGS data from any applicants up till now. And I have been working in NRA 10 years. I don't know why, but maybe due to lack of applicant experience regarding the use of this advanced method. And I think it will need a long process of method development, validation before implementation. And I think industry need more support or training regarding the use of these advanced methods.
Thank you very much. Maybe you didn't receive data yet, but it will be interesting to hear if you had any discussion. Did manufacturers approach you with a question about the alternative replacement of in vivo assays or in vitro? So has this discussion taken place or not yet?
Not yet.
Thank you. Okay, then let's move to Bangladesh to hear from Hamida. How are you dealing with these issues that your colleagues just described in their countries?
Okay, thank you. Can you hear me?
Very well.
Thank you. Thank you for inviting me in this conference. Actually, this NGS technology is very new and it's not in practice in our country. But in our country, I am from NRA, but our manufacturers are more advanced than NRA. I am telling the truth, actually. Our some biologicals and vaccine manufacturing company trying to adopt this NGS technique. And in that case, we generally visit our facility and evaluate their documents, their module, clinical trial data, and also we evaluate the testing result from abroad, from foreign country, foreign lab, which are WHO pre-qualified lab. On depends on reliance, and this reliance techniques is supported by our law. As NRA, we are trying to we have planned to implement this NGS technology in our lab. In our laboratory, we have a cell culture facility and well-equipped microbiology lab and vaccine testing lab, and also in vivo testing lab. But we are trying to replace in vivo testing with in vitro testing. So as NGS technology is more advanced, we will welcome NGS technology in our laboratory, and we will try to implement this technology in our laboratory. Thanks from my side.
Thank you very much. And also thank you for sharing the information about manufacturers being more advanced. This is exactly what we asked manufacturers many years ago, to really take a lead and to come up with a technology that would then require also building the expertise among regulators. So at this point in time, we still see that probably further encouragement is needed. And we're also thinking what kind of training or technical support to regulators would be useful or would be most suitable at this point in time to facilitate this discussion between manufacturers and regulators. Even before you receive the data, it would be useful actually for manufacturers to come up with a proposal to discuss this with regulators, because this early dialogue can actually save a lot of time on both ends. And then there will be no waste of time when they come up with the actual data. It will be easier for you to review. But maybe I think other panelists may have other suggestions or questions.
I'm just picking up a little bit as what concerns me and what we also discussed in the European regulatory field. It is what you mentioned, batch-to-batch analysis or comparison. So the idea was I have a critical product, I want to be sure. So I do for a while the testing with both methods, let's say the in vivo plus the NGS or the in vitro plus the NGS. And if something is the difference, I should be cautious. If there's no difference, everything seems fine, consistent. But I really would be cautious with this approach, because contamination events are very rare. And if you have a negative batch, or if you have 3 negative batches, both methods will be negative. And in the end, it tells not too much. And just to tell from the experience, I remember this with a plasma-derived product. The company did a clinical trial for parvovirus B19 transmission. So they even looked at the patients, the recipients, and even looked having young children that have not been infected, and did a nice clinical study showing that the batch that they had in the clinical trial, of course, did not transmit. And this was true. This was nice. And they told us, yes, we have clinical confirmation that our product does not transmit parvovirus B19. This was true until the first contaminated batch we had the transmission. So I'm really wondering whether this, I know we are all sticking to these 3 comparability batches and so on, but for these rare contamination events, I'm really wondering whether this strategy would be helpful.
Yeah, I agree. I don't think it is very helpful. And coming back to this comparability question, I think we said there is no need for head-to-head comparison in the ICHQ5A, and you mentioned very well this morning, Johannes, that there is a possibility to look at published data, already prior knowledge in a company, for example. And I think, you know, of course, it's compendial tests, and there is a statement in the general notices of the European Pharmacopoeia that you don't have to validate, but it doesn't mean you do not evaluate the performance of your test, because you may want to evaluate the performance of your test because of your matrices. So I am aware that in the companies, you have some data using some model viruses, limited data. It's not a full validation, but it's like a verification or qualification that is done to evaluate the performance of your in vitro method. And if you have still the references which have been used to do that, you can reuse these references to look at the NGS performance with these model viruses, in addition, of course, to the full validation package, which is required. So I think it's not to say there is no comparison that is needed. It's just to say there is a possibility to compare if you have some materials which have been used from previous studies to evaluate your in vitro test, for example, or your in vivo test, of course, but in vitro in particularly, generate some data to do this comparison. But I agree, testing in parallel 3 batches with both methods will give you nothing, because at the end, it's always negative. And I just want to come back to that, because I think this morning we had this very nice presentation from Yusha about this survey that you have conducted also, how it was detected. And in fact, for the European Pharmacopoeia, and I think Wenael can also comment on that, when we revised the chapter 2616 and 523, we have also conducted a survey. Of course, it has not been disclosed publicly, but to support the revision, we looked at the cases of contamination, how they have been detected, and we have done a survey from both the manufacturers, the CROs in Europe. And it was very clear that there was nothing which was detected with the classical methods, at least in vivo. Some with in vitro, but never alone for in vivo, for sure. and some with in vitro, but most frequently it was with molecular methods. So this was also the driver for the revision, and I think it's very important to keep that in mind, is that if you do in parallel 3 lots, you will find nothing.
Maybe speaking about survey, because so for when we elaborated the chapter 2641, we also had a large survey with the stakeholders. So I thought I wanted to mention that because I think there was a huge turn up for the survey with stakeholders. We had more than 20 responses, which I think is very good. It's a very extensive survey, and it really helped us to build the chapter and to inform what we describe in there with the best practices. So for each step of the workflow, and then the controls, and there is also the validation part. So I think it was really a critical element for building our chapter 2641. And this led us to what we have today. And I think it suggested also that there is a very good NGS community that is really dynamic and support us with the data to build that chapter.
One last question also to the CAAB consortium. Yes, I was wondering, you did not yet include the veteran and medicinal product from the veterinary field, but there's a lot of experience with contamination events, and whenever I want to learn what can go wrong, I would go there. And certainly those colleagues have to operate in much more financial constraints. So the safety measures, so it's really a tight balance. And so there's a lot of experience. You can learn a lot of virology from the veterinary field. So are there any plans to adopt or to look at the experience? Because it might be also useful for the human field to learn what can go wrong and what is necessary.
Yeah, I mean, that's something I think we would love to do. Obviously, the study that we did is contained within our CAACB membership, so we would require those companies that are doing that sort of work to join our membership in order to do those studies. But assuming that that's something that they're interested in doing, that's certainly something that we at the consortium would love to look at.
Johannes, what you mentioned as a kind of a constant learning, I think it might be useful for regulators who are preparing themselves, to introduce this, just to maybe provide some kind of brief overview how you got into that. And we have a lot of tools and a lot of kind of useful experiences today, but still this introduction of HTS and replacement of especially in vivo assays is a very slow process. So from your experience at Paul Ehrlich and maybe in the European context, if you can describe and maybe give some advice what to do and how to start getting into that, and maybe then Michael, if you want to say from Canadian perspective, but
very simple message. Don't be too shy. It's just to start. Yes, and yes, of course, we should share all the knowledge. But I think that there has a lot of and that's of course there's a great value in the CCAB consortium and probably there might be much more contamination issues around than those reported. I even know some, but I'm also probably not the first person where everyone runs and here I found something. Yes, But I think really it would be good to have the publication. I like all this activity here to share the publications, also the working group from the Adventitious agents, PDA group to publish everything, and then just to start and a little bit trust in this, what we sometimes call external prior knowledge. So what has been published and is there.
Should I comment on like sort of replacement of animal tests in general, or like the ease of
what is
giving
the
some-
situation in Health Canada and how you see these moves and changes?
Yeah, so we're in an interesting position because we do interact a lot with 3Rs and replacement of tests, but legally we're not bound to reducing animal testing. That's not part of our situation. But for the most part, we want to get rid of animal tests because they tend to be terrible. There's few instances where an animal test is superior to a cell-based assay and things like that. So things like removal of general safety tests. There's other things where basically it's been easy because as soon as a manufacturer comes in with the submission, we just basically approve it on the principle that we don't think something we wanted anymore, kind of thing. And so when it comes to replacement of an animal test or even in vivo test, what matters most is performance of the assay. So we don't, put in principle, that's obviously part of the principles too of reducing animal testing, is that the tests have to be at least as good as or superior to that. And our experience has been pretty good overall, I think, of manufacturers coming in. It's always this delicate balance of claiming that we're always open to everything, and then manufacturers, we almost put it on the manufacturer to say, no, you need to propose something to us. But of course, there is that trepidation of like, are they going to accept it or not? As soon as they submit something, we might be very judgmental. And I have to say in general that my groups were very open to pretty much any proposal. And we have very non-prescriptive regulations. We don't really publish our own Canadian specific guidance documents. We work internationally. So basically, by law, we could accept almost anything that's scientifically justified, but obviously there's limitations to needing harmonization across a lot of different regulators. So that's really one of the biggest burdens, I would say, is just the burden on regulators of needing harmonization across so many jurisdictions. And so we always try to offer if they can meet us.
Maybe I just want to comment one thing. I think it's true that in Europe we have a directive regarding animal reduction, but I think here the main driver for introducing NGS is not only the animal test reduction, it's the scientific basis of what we have seen during this conference and the previous conferences, is that you may miss some contaminants with the traditional assay. And I think it's a question of risk assessment. And I think risk assessment is also very present in the European Pharmacopoeia chapters. And the idea is not to replace and to remove animal testing just for the pleasure of removing animal testing. It's to secure the risk assessment aspect and to cover the gaps that we have with the traditional methods. And I think it's very important to keep that in mind, is that traditional methods are not sufficient.
I think I'll just mention too that even among some reviewers, there's this perspective that NGS has a sensitivity issue and that in vitro assays, PCR would be superior, even some of the animal assays. And it's about educating them that no, we're at the point now that really NGS is as good as PCR and the in vivo assays are really not ideal. So
I think we're going to have to close this very productive, active session. I had one more thought about the in vivo assays that I wanted to share, but I'm not going to say it because that's going to take up more time. Maybe over the next day, we can, I'll bring it up again. And it's not to why we don't need to use them, but as to why they may have been used before. But I've been struggling with that question. I think I have an answer. But anyway, so we have to go, I think, to the next session, which we're already delayed. So please, don't, we'll continue this, over the next day or so, especially we have a final panel discussion also. So with that, thank you very much to the panelists and to the audience and to our guests online as well. Thank you so much for your participation.